Very good morning to one and all present are here. I welcome you all to the second day of virtual international conference on clinical and applied microbiology on 27th and 28th of April 2023, organized by BioLEAGUES, Chettinad Academy of Research and Education, our academic partner, and United Innovators, our research partner. I would like to proudly introduce and welcome the keynote speakers for today, Mr. Neelakanta Banerjee, Head Microbiology Department, Aurobindo Pharma, USA, East Windsor, New Jersey, United States of America. Dr. Priyadarshini Shanmugam, Professor and Head, Department of Microbiology, Chettinad Hospital and Research Institute. Professor Dr. Kailas Dasharanth Sonavane, Head of the Department, Department of Microbiology, Shivaji University, Kolhapur, Maharashtra, India. I would like to welcome and introduce the session chairs for the Day 2 conference. Dr. Mukta Sharma, Professor and Deputy Dean, Microbiology, IIMT University, India. Dr. Srijit Das, Research Scientist, University of Iowa, United States of America. Dr. Atreyi Ghosh, Professor, Microbiology and Biotechnology, Sister Nivedita University, India. I welcome and thank each and every participants and welcome you all. Now, I would like to welcome the first keynote speaker for our Day 2 conference, Mr. Neelakanta Banerjee, Head Microbiology Department, Aurobindo Pharma, USA. Sir, now the screen is over to you. You can take over the session.
Thank you, Bhargavi. Can you please share my presentation today?
Yeah, sure, sir.
Or I can do it from here also. Hello, good morning, everyone. So this is very night in the US, so that's why my sound is little bit, down now. It's 11 o'clock, 11 p.m. And I always like to, participate wherever microbiology work or some innovation talk is going on. Yes, the name micro does not mean there is a small thing. The full of world is filled with the microbes. In present scenario, COVID is shown everyone what is the power of microbes, the good way and bad way. I'm not going to that details because everyone having their own time, but I want to because this is applied microbiology. I need to speak a little bit about the application. Everyone knows there is a many application in micro, but especially pharmaceutical industry is a big important role play microbiology. One of the important thing contamination control strategy development pharmaceutical manufacturing. There is a many guideline now, talk on that, especially EU Annex guideline and PDA Parental Drug Association. That is a technical report series 90. Talk about contamination control strategy. So if you see the previous guideline, 2007, the guideline only to 16 pages guideline. And now 2021, 22, the guideline is updated and it will, 52 pages plus glossary. There's a lot of, new addition, new rules come, and across the pharmaceutical industry, they're supposed to be follow that regulation to, do the quality medicine. So what is the key highlight of this guideline? That guideline talk about the contamination control strategy. There is a new revision on, classification and monitoring. May I audible to everyone or any problem over there?
No, sir, you are audible.
Okay. There is a single use bag system come. Many important thing, very detailed. If we go, it will, never go within half an hour time session. So I will like to, overview just. So any contamination control strategy implementation, there is, strong foundation is very important. So this presentation is basically, I will, touch few points. That is a strong foundation, contamination control strategy, classification, qualification, monitoring, single use bag, isolator system that is called, that is come under various system and other advanced technology. So you can see in the strong foundation, that strong foundation means it's, aseptic process and aseptic product where we are manufacturing. That is very important. And quality culture. So if you see in the pyramid, top of the pyramid, there is a mentioned sterility test. But we only achieve that sterility if our foundation, if our pyramid is, strong. So pharmaceutical manufacturing facility, pharmaceutical quality management system is very important. One of the pillar, facility design, equipment design, personal training, gowning, aseptic technique, and then come the classification and qualification. Then important thing, cleaning, disinfectant, particle, and process monitoring, sterilization, and sterile filtration, media fill, and sterility test is the final assurance. So if everything goes well, then only we can say yes, our product is sterile, our sterile medicine go to needy patient, and patient will get the right medicinal drug. So it's very important. Second thing is contamination control strategy. But before that, you have to know the cost of contamination. Cost of contamination is is not easy. If there is any failures, that failures, the final cost, not only the dollar or money, it very important, someone life that cannot be reversible. So that is the final patient safety, product safety, and then come the organization credential and their brand value. So that is the cost of contamination. But control strategies, it depends on how you design your facility, microbial control, contamination, pyrogen, endotoxin, particle. It's depend on everything, how management work, how aseptic behavior follow. So education is one aspect, they give you the provision to come, but honesty, integrity, and transparency matter in this field. And new idea, following guideline, that is very important. So microbiology, very important aspect in especially in pharmaceutical industry, sterile manufacturing. And now next generation drug also come used for the cancer drug research, that is called cartesian therapy. So very important part, very important aspect, vast of area. If I say if world is filled with air, one more thing I need to add, filled with microbes. Not only the world, out of world also. So importance of microbes you understand. Next, I need to tell the elements of consideration during contamination control strategy. What are the elements? What I already tell. My left side slide is mentioned, design of plant and process. If you not properly design any area, design not only the architect point of view, design point of view of classification, grade A, B, C, D, ISO 5, ISO 6, ISO 7, ISO 8, CNC, as per US FDA, it is a class 100, 10,000 and 100,000. So different classification is needed. Or sterile drug manufacturing under class A, ISO 5, and class 100, grade A. There is a microorganism acceptance criteria is 0. Initially it's claimed less than 1, now it is 0. So you understand it's not easy to achieve that criteria. So proper design is very important, cascading is important, aseptic behavior is important, premises, equipment qualification is important. Personnel, those are the personnel involved in this process, how they are aseptically, follow their behaviors, techniques, procedure, utilities. All utilities also important, play important role, whether we are using qualified utilities. Is the utilities under, WFI, purified water, pure steam, compressed air, nitrogen. We are using raw material, all the incoming material, raw material, pharmaceutical substances. It need to be properly tested. Any container need to be, checked. Primary packaging material, secondary packaging material, everything need to be tested through micro point of view. Chemical, definitely micro also, especially in sterile. Supplier, vendor, approval, outsourcing activity, if you've done anything, that also need to be monitored properly. Risk management, risk assessment, process validation, sterilization process, preventive maintenance. These all are one topic is taking 1 to 3 days to explain. Cleaning, disinfectant, monitoring system. I can say sterilization, depyrogenation, filtration, radiation, gamma irradiation, many things. Trained evaluation, investigation, proper corrective and preventive action. And finally, when everything following come the governance that I presented in my right side of the slide. So that is important, very important role. Every department play important role, whether it is microbiology, whether it is a quality control, chemistry, manufacturing, quality assurance, validation team. But I always say it's a quality unit. It's not the quality assurance one person is give or one department is give the assurance of any drug or medicine. So it's a quality unit as per US FDA. So that's play microbiology is very important role in sterile pharmaceutical industry. Same thing. My next slide is classification and clean air equipment. There's a many upgradation come as per new guideline Annex 1. That critical area classification must be requalified that area, grade A, B every 6 months, and grade C and D every 12 months. So what are the tests? That HEPA filter integrity, air velocity, volume, pressure differential, recovery every 6 months. These are the tests is importantly. you have to be requalified. Classification as per ISO 14644, part 1, table number 1 is given, same thing following 5 micron and 0.5 micron. So these are the many things come. And monitoring aspect, environmental monitoring, total particle, viable, non-viable, is a continuous monitoring, temperature, humidity. These all are aspects give the quality medicine. You can see there is a guideline having, at rest, in operation, during validation. Hello. Yeah. So you can see many aspects come of the guideline for the routine. How you do when you do the validation, there is a not specified any address condition acceptance criteria. If you see in the routine, there is a acceptance criteria come for the particle 5 micron. So these are the things is very important for the new addition in Annex 1 guideline that must be followed everyone. Next thing is the viable point of view. Initially, that guideline saying less than 1 person, less than 1 CFU colony can accept it in ISO 5 or grade A. Now it's changed to no growth. No growth means absolutely no growth. So that is some presentation change. But you can see the stringent limit. Grade A, no growth. Grade B, for the air sampling, 10 CFU per meter cube. Grade C, 100 CFU per meter cube, and grade D, 200. Same way, settle plate 5, 50, 100. So you can see there is a many things come and is very difficult to follow. So it depends when there is a good classification, classified environment, people behavior, upcoming material tested, then only people can follow. This limit can achievable. This is the key some environmental and process monitoring requirement come for the grade A total particle and viable monitoring is fully during all critical operation. It's start to end. That the initial also now also all sterile manufacturing facility they are following end-to-end monitoring. But I always prefer do the isolator system adoption that give you great compliance in terms of FDA point of view, contamination point of view, because human is a great source for any contamination. So using isolator is one of the best. Nowadays you can avoid many non-conformance by using isolator. Next, same thing I tell that how you control the low bioburden drug substances, process control. Same thing your facility that HU, pest control, flow of personnel, material, waste. So unidirectional flow of material, personnel, equipment. All equipment should be classified, qualified. All process must be validated. I can say here when you are using any manufacturing equipment, when you are doing filling under close wrap or isolator system, you must be followed, and that must be cycle must be validated with the BI, biological indicator. Cycle development, cycle validation, all are important aspect of validation point of view. All material must be tested, release. Approved vendor required to use utility must be routinely monitor, validate, and use. Personnel obviously the train. Then only you can able to microbial control. Now, one more, important thing added this guideline that is newly added, that is called single-use system. Single-use system, single-use bag. Somebody call, is a P-use, pre-sterilized single-use bag, one-time use, and through some of the product, they are, metal interactive. They are react with the metal. So that time that single-use bag is very important. So how you validate that system? How you assure that sterility end to end? This procedure come in this guideline. A very important filter integrity pre and post. So these are the thing is mentioned this guideline, and these are the part of contamination control strategy. And I like to tell various system that is called isolator. Isolator having two type of isolator. One is the open isolator, closed isolator. Open isolator, critical zone grade A, unidirectional, and background is minimum grade C requirement. But if you see the closed isolator, the critical zone grade A, no needed any unidirectional, airflow, but background is minimum grade D. And VHP, cycle development, cycle validation, everything is happening with 35% hydrogen peroxide, vaporized hydrogen peroxide. VHP is a vaporized hydrogen peroxide. So it's a surface sterilization act, nicely, eliminate all the contamination inside the filling area, and throughout the process maintain the aseptic condition, and you can easily achieve grade A requirement. So restricted access barrier system people are using. I always recommend it is very advanced and costly. Definitely, isolator is more cost is not everywhere important. Where is quality is matter. So sterile businesses, sterile injection medicine, quality is final. So quality give you everything. So you can think what system is supposed to use. Yes, what I'm telling that single use bag about that requirement very clearly mentioned. When you do that single use bag validation, you have to be see the qualification. But in terms of microbes, you have to be, for the sterility and endotoxin level, assurance also needed end to end. So these are the requirement coming new guideline. But it is very, aseptic observe program. It's very important how you do it. Qualified operators that require identify and train them frequently, continuously. Observe aseptic behaviors. If not properly, again you train. But its continuous observation is required. If you say phone number, report to operators and get feedback, what difficulties they are facing. That you check, that you improve in process also. So that improvement is mentioned over there, and analyze and train, observe how many times they are doing any failures. That is important. And one program require to implement their procedure. Then only you can maintain that aseptic behaviors. So that procedure also mentioned over there this guideline. Final took out of this guideline basically how you maintain an aseptic condition, sterilization, disinfectant behavior, filter, cleaning. All our thing is important. Continuous monitoring, particle, drain quality. So these all are important to maintain aseptic behaviors and contamination control strategy. This guideline is very important, and already the deadline is 2025, 5th August 2023. Except there is a lyophilization section is there that they are given regulatory given a loose part, automated loading and unloading system 2000 up to 2024. The rest of the thing already effective. Those are the facility and new facility. They are effective since August 2022 when this guideline are coming. But the overall thing I want to tell that is yes, microbes now play everywhere important role. And initially in microbiology, especially in pharmaceutical industry, people are consider microbes or microbiology department as a name of micro small. I always try microbiology is not the dot anywhere. It's the heart of the any plant, any good plant, their heart, any sterile plant, their heart is the microbiology department. And now it is the brain of microbiology department for any of the aseptic pharmaceutical industry. If you send FDA citation, observation, every company having their problem in microbiology, because I always believe, it's happened through the, not proper knowledge, ignorance, and not given importance. They think people are thinking microbes are not visible, so they are not capable. But it's absolutely wrong. Human think human are very intelligent, but no. I believe no. Intelligent. So think about the microbiology new implementation and all together work to take this battle and do everybody best. Good wishes everyone. Thank you for calling me. Any question for anyone, please?
Thank you, sir. That was an informative session. Participants, if you have any questions, you can mention your queries in the chat box, and sir is ready to answer your queries. I repeat again, participants, if you have any queries regarding this session, you can mention your queries in the chat box.
Thank you.
Sir, I hope participants are clear with your session.
Thank you.
Thank you, sir. Thank you for joining us today, and thank you for your valuable information to the participants.
You're welcome. You are welcome. Thank you.
So we are going with the second session for the day. Dr. Priyadarshini Shanmugam, Professor and Head, Department of Microbiology, Chettinad Hospital and Research Institute. Ma'am, the screen is over to you now.
Okay. Thank you. At the outset, I'd like to thank the organizing team, both from BioLEAGUES as well as my team here at Chettinad. And I hope you have a fruitful session. This talk will be on emerging and re-emerging infections, an overview. And I am Dr. Priyadarshini Shanmugam. I'm the Professor and Head of Microbiology at Chettinad. And Microbiology Department at Chettinad runs diagnostics. We also teach the medical students, and we're also actively involved in research. So I've been here for over 18 years. I joined here as a junior faculty and assistant professor, and then I actually made my way up to the head of the department. And I could say I've grown with the institute. The institute has given me numerous opportunities for improving myself at the same time while contributing to the Institute. Actually, I have completed my undergraduation from Government Keelpok Medical College, my post-graduation from Madras Medical College, Chennai, and I also completed my PhD from Chettinad Academy of Research and Education. That is the university to which our college is affiliated to. So now I've finished off with the introduction. Now I'm going to tell you about the synopsis of my talk. So actually, I will be talking to you about emerging and re-emerging infections under the following headings. So what are the definitions, relevant definitions? Then what are the factors that contribute to emergence? Then certain examples of EID. EID stands for emerging infectious diseases. Certain case studies on SARS, Nipah virus, H1N1. And then how do we go about with the prevention and control of these emerging infections? So before that, I just want you to see, this is the global burden of infectious diseases. Okay. So annually throughout the world, we have about 57 million deaths occurring throughout the world. And out of which 15 million, that is more than 25% of these deaths are due to infectious diseases. So we now know that infectious diseases are a big burden on the healthcare industry as a leading cause of suffering, that is morbidity, as well as death, that is mortality. Okay. And among the infectious diseases, these are the common causes of deaths, that is respiratory tract infections, then HIV leading to AIDS, diarrheal diseases in the developing countries, tuberculosis again in the developing countries, numerous vaccine preventable childhood disorders, malaria, sexually transmitted diseases, meningitis, hepatitis B and C, you know, they lead to chronic liver disease leading to cirrhosis and carcinoma, tropical parasitic diseases, dengue, and other infectious diseases. So you have a chunk of infectious diseases which lead to deaths. And WHO is the World Health Organization, which does a lot of screening, okay? And they keep monitoring for the different outbreaks and all that. And it was seen that in the 2007 itself, WHO has warned that numerous infectious diseases are emerging at a rate which is much higher than before. So since 2007, you have diseases which are coming, new diseases which are coming up at a very rapid rate. So that was what they're giving the warning. They gave the alarm bell. And since the 1970s, so that's about, you can say about 50 years before onwards, about 40 new infectious diseases have been discovered, okay? Like your HIV and AIDS, that was in the 80s, SARS-CoV-1 first, Ebola, avian flu, and swine flu are a few examples. So all these diseases are high threat and they have pathogenic diseases which have increased in the recent years throughout the world. And one main factor causing this is because people are traveling more frequently, because there's more accessibility, then they're traveling greater distances than in the past, and they're traveling more frequently than in the past. So this is a main factor by which the diseases can spread rapidly between countries and continents, okay, and cause global epidemics. And this was the picture which we saw in SARS-CoV-2, which was the most recent pandemic that we came across. And it has also been seen that most of these recently emerged
system.
Doctor, I think others, I want others to mute yourselves. Suja, please mute yourself. And about 75% of the recently emerged diseases all have a zoonotic origin, meaning that they are originating from animals. So zoonoses are infectious diseases which spread from animals to human beings. So in this slide, I have put up certain papers, okay, and all these are publications on the latest outbreaks. Okay, some of them, like you see, there was a Nipah virus outbreak, okay, in Kerala in 2018, which you all would have seen. Then there is an emerging pathogen, cryptosporidium, has been reported. So like that, many diseases have been reported, okay. And we also see that there are a lot of multi-drug resistant organisms evolving, okay, and they are emerging. So these are, and there are innumerable publications on these emerging infectious diseases, okay. So now what is exactly an emerging infectious disease? So they're called as EID in short. And these are infections which recently appeared in a population. And these infections whose incidence means number of new cases. So the incidence or the geographic range, that means over how many states or over how many countries or over how many districts, that is the range, okay? That geographic range is rapidly increasing, or it is threatening to increase very shortly, okay, within a short period of time. If it is going to, if you think that it's going to spread rapidly, or it has already rapidly spread, okay, it has spread out over its geographic boundaries, then you call it as an emerging infectious disease, okay? So number of new cases are high. So what's a re-emerging infectious disease? So these diseases or infections were known earlier, okay? But then we have instituted proper infection control measures. So as a result of which the levels fell low, okay? Because they had been previously controlled. But then what happens? The infection control measures had become slack. You see, that's what always happens, right? We become complacent. So again, these diseases start showing an upward trend in the incidence or in the prevalence, okay, worldwide. And they are appearing in areas where they were not previously found, okay? So one is showing an increase in the incidence or prevalence worldwide, or showing up in new areas. And these have risen to become a significant health threat. So these are certain articles which you can see, like the articles on polio, which showed that the disease has re-emerged after 6 years, okay, in Ethiopia. And another article shows that polio virus returns to the UK after nearly 40 years. So one was published in 21 and one article was published in 22. And this figure has actually been taken from an article. The name of the article I have put up on the left side. It's the challenge of emerging and re-emerging infectious diseases. So actually, this one of the co-authors for this article is Dr. Anthony Fauci. He was the director of the National Institute of Allergy and Infectious Diseases, okay? And he was the one who was playing a vital role in this infection control during the COVID pandemic in the US. But I think now his term is over, or his tenure is over. Okay, so in this you will be seeing, this is the map of the world, okay? And you'll be seeing numerous red dots and blue dots, and then there's one black dot. So the red dots are the ones which are the newly emerging infectious diseases. So some examples I have given here, okay? They are marked out here. And then the blue dots are the recent re-emerging or resurging infections. You can see here, like typhoid fever. And then here there is a diphtheria re-emerging and all that. And then there is one black dot. That is a deliberately emerging infectious disease. And the reason for that is bioterrorism. So we'll just touch upon that also a little later. So newly emerging, then there's another term re-emerging, and then a deliberately emerging. So this is the timeline of appearance or occurrence of the different emerging infections throughout the world. Okay, so you can see right from 1975, we had parvovirus B19 emerging. And then you have protozoa, then you had viruses and bacteria. So everything in order has been given. And the more recent ones is influenza in 2009. I'm sure most of you would have been, who would have experienced that fear during the outbreak of this H1N1 influenza. And then we had a MERS outbreak in 2012 to 13. Okay. And then you had a severe fever with thrombocytopenia symptoms was there. And then in 1920, or rather December 2019, and then 2020 onwards, you had the COVID-19 pandemic and the SARS-CoV-2 being described as the emerging pathogen. So whatever I had shown in the previous slide, like this is the timeline and this is the organism wise. So you'll have emerging viruses, emerging parasites, emerging bacteria, and emerging fungi. And among the emerging bacteria, it's the same bacteria, but they may come with some drug resistance, and they are also considered as emerging pathogens. Like you have the multi-drug resistant and the extremely drug resistant Mycobacterium tuberculosis, methicillin resistant Staph aureus, and vancomycin resistant Enterococcus. So we had so many people presenting on all this yesterday, right? And then you have the emerging fungi, like the non-albicans Candida. And among the Candida, the most dangerous one here now is Candida auris, which we have many isolates. Even in our hospital, we have seen, and there were increased in the number of these cases during the COVID-19 pandemic. And again, you have the re-emerging fungi, the zygomycosis, okay, which was called as the black fungi, again, during the COVID pandemic. So these are all the examples of emerging viral, parasitic, bacterial, and fungal infections. And this is the timeline of the emerging infections in India. And again, it started with 1992. You had Vibrio cholerae, 139 strain, going on to plague, diphtheria. So the first three are all bacterial, and then you had a viral outbreak, the Nipah virus, and then again plague, and then these are all viruses, the Chandipura virus, the Chikungunya virus, influenza, Crimean Congo hemorrhagic fever, and all that. So this is the timeline of emerging of the infections in India. And now let's go on to the epidemiological triad of disease. And we all know that infections do not occur from a vacuum. So you need to have a main thing, a host, a susceptible person who will harbor the organism and in whom the organism will multiply and cause the disease. Then you have an environment, okay, from which either directly the host can get infected, or from the environment through a vector, the host can get infected. And you have an agent. The agent also is the organism, the infectious organism, which is going to cause the disease. And it is different from the vector. The vector is just like a carrier of this agent, and it spreads the infection to the host. The agent also can directly infect the host, okay, or through the environment, it can go and infect the host, or through a vector, it can go and infect the host. So this is the triad of disease, and anywhere you can break the chain, okay, you can control the disease, you can prevent it from spreading. So that is why it is important. You need to know what is the vector for an infection, what is the agent for the infection, and how the environment plays a role, and how we can break the triad or break the chain somewhere. Okay, right. Now, we spoke about the different emerging infectious diseases. We saw about the definition of the emerging and re-emerging diseases. Now, we talked about the infectious agent. So what are the factors, or rather the agent factors that contribute to emergence? So one thing is the pathogenic infectious organisms have evolved. Okay, the microbe has adapted to the environment, so it has changed, and this change can be a series of mutations. So just one mutation itself can cause a new organism to emerge, or a series of mutations and series of changes, in the organism, which has led it to evolve and cause diseases. The development of drug resistance. Another thing which you can add to it is the resistance of vectors to pesticides. Like for instance, you would have seen that in our country itself, you would have seen that these mosquito control measures will be initiated by our municipal authorities or the corporations. And you will see that still the mosquitoes will be flying around even after they do the fogging and go, because it has developed resistance to the pesticide. Okay, so many years, over so many decades, they have been fogging with the same chemical compound. And because of that, the vectors have become resistant to it. And I already told you that drug resistance also, you can take it as an emerging bacteria. Drug resistant bacteria, you can take it as an emerging bacteria. So this is the timeline of development of resistance. We are taking one example, Staphylococcus aureus. You can see that during the Second World War, a lot of people, a lot of lives were saved because of the discovery of penicillin by Alexander Fleming. But very soon after penicillin was discovered, the penicillin resistance followed. And this was also predicted by the same Alexander Fleming. And then they developed a drug called as methicillin to overcome this penicillin resistance. And then Staphylococcus aureus, kind of found out a way by which it could overcome the methicillin resistance. Then a higher drug was discovered in the 90s, vancomycin. And now you have resistance to vancomycin by enterococci, and you have vancomycin intermediate Staphylococcus aureus also coming into the picture. And very soon you will have the vancomycin resistant Staphylococcus. I think we do have some strains, but still, hopefully, thankfully, it is rare. Okay. So in drug resistance, what are the factors that contribute to the emerging drug resistance? So this I have just taken slightly away because I want to stress on the drug resistance. Wrong prescribing practices. Okay. You're not writing the proper dosage. You're not writing the proper duration, okay, of treatment. Okay. And then that will lead to the organism surviving and then developing resistance. Okay. The second thing is the patients are not compliant. So you have prescribed properly, but your patients are not taking the drug correctly. Counterfeit drugs, which may not have the proper amount of the antibiotic. Use of anti-infective drugs in animals and plants. So what we do, we give it as a fodder for the animals. So everywhere the drug-resistant bacteria will start surviving in the gut, and that is how it spreads from animals to human beings. Okay. So this is a picture which shows this natural selection of resistant bacteria. So in any given, a group of bacteria, resident bacteria which are present, okay, what happens is you'll have 1 or 2 resistant strains there, okay, in the bacteria which cause an infection, okay. And all the yellow dots are the sensitive, drug sensitive to a particular antibiotic. They're sensitive bacteria, and these are all the resistant bacteria. So you give an antibiotic to which your bacteria are sensitive. What happens is all the drug sensitive bacteria, most of them, 99% of them get wiped away. But your drug resistant strains are going to be surviving, and then they multiply. And finally, that population gets replaced by a population of drug resistant bacteria. So this is only known as natural selection. And what are the consequences of antimicrobial drug resistance? So one is, right, your patient gets admitted to you for undergoing an elective surgery. And then what happens? The wound gets infected. That becomes a surgical site infection. So you cannot discharge a person with a surgical site infection. That is going to be a wound that is discharging pus, right? He came for a clean surgery, but he's going back with an infection. That's not correct. So this will prolong the hospital stay. Sometimes the infection may get the better of the patient, may lead to a higher death rate. And then, drug-resistant bacteria are susceptible only to the highest antibiotics available, which are very expensive and toxic. And because of all this, it leads to higher healthcare costs. Okay. So now we had just deviated a bit from the causes of emergence among the agents, and we discussed about the antibiotic resistant strains of organisms. So now what are the factors contributing to emergence in the host? That is, here we can take it as our human beings. So our demographic change, suppose we go and inhabit areas, new areas, where you have an increased contact with animals and with the natural environment. Okay, that means nothing. You are just going more closer towards the forest. That is an example. Okay. Second thing is abnormal behavior. See, normal behavior is different. Some, abnormal sexual practices, drug abuse, sharing needles, body piercing, tattooing, all this can also cause a susceptibility to infection. And due to changes in our lifestyle, we can become more susceptible to infection, or we may get immunosuppressed either as a result of taking medicines or under stress or some other lifestyle modification we have done, which makes us more susceptible to an infection. Nutritional changes, right, the food that we eat now, even if you keep it in the fridge for about a month, it doesn't get spoiled because that some chemical has been added. So more use of pesticides, more use of chemicals, that is changing our nutritional status. So that can lead to this emergence of newer organisms. And then poverty, social inequality, these are all social factors, okay? And this is further triggered by some wars you would have seen in Ukraine, some civil unrest. What happens whenever a war is there? Refugees are created. People are running around in search of food and housing because they have lost their own houses. They don't have a proper source of food. So there's a shortage. People are crowded into areas. So there is an increased density of living. So among them, what happens? Disease spread is faster. And some people, for the fun of it, they undertake some outdoor activities like trekking and swimming. And again, here it takes them closer to nature. But there is a risk also involved there. They might be exposed to some newer pathogens which are there in the forest, and you might pick it up and come home, right? And other host factors are globalization. This I mentioned in the first 2 slides itself. Increased travel, international travel. Then, these mosquito eggs, when shipments of tires, for vehicles, when they were being shipped, water used to collect inside it. And then you have eggs of these mosquitoes breed. And then because of that being laid there, and then the mosquitoes breeding there. So this happens in long distance travel. Another main feature is this wild animal trade. Okay. This was the case in the case of monkeypox, West Nile, okay, and SARS. And other host factors that lead to emergence are our agricultural practices like pig farming. I'll discuss the case history. So this is seen in Nipah virus, then goose farms for this West Nile virus and so on. So there's a breakdown of the public health measures. Okay. So usually the municipality or the government takes care of this public health measures like chlorination, vaccination of the citizens, vector control measures. So anything breaks down, it might lead to emergence of the infectious diseases. So breakdown of vector control, one example is when there is an increase in the abundance and distribution of this Aedes aegypti mosquito, suddenly there was a high incidence of the number of dengue hemorrhagic fever cases in the US. Okay, so that was a thing. And then lack of political will, this you would have seen in some countries which do not report the global infectious diseases, okay, for political and economic reasons. So we don't know what is going on inside their country, okay. And then improved diagnosis. You're able to diagnose it as an emerging disease because you're able to first detect it, because you have improved diagnostic techniques like molecular methods and all that. Of course, technology, industrialization. Industrialization, again, what you're doing, you're spreading your industries, you're breaking down the forests. And because of that, you know, the animals do not have a home, so they come into the cities. So this is what happens. So these are all the host factors. And another thing I told you, more than 75% of all the emerging infectious diseases are due to they are zoonotic in origin. That means always there is a spread from an animal to a human. Okay. And so this slide is an important one. See, this is how the zoonotic diseases emerge, okay, from different animal-human interfaces. So interfaces where all humans and animals can come in contact so that a disease agent gets spread. So one is live animal markets. So this is what we saw in this COVID, okay, in Wuhan, in China. So the animals are being sold there live, okay, or they might be sold either legally or illegally. This led to the spread of SARS and avian influenza, intensive wildlife farming, okay, in wildlife hunting. And then, of course, from domestic animals also we can get the influenza, Nipah, Hendra virus and all that. So all that human beings who come in close contact with any one of these activities or animal related activities can get the infection. So they will be the index case or the first case. And then when they get hospitalized, the unfortunate healthcare worker will also get the disease, and their relatives will also, the family members, the contacts will also get the disease. And that's how it then later it spreads from person to person. Okay. And what are the factors, the environmental factors that contribute to emergence? So climate change, okay, changing ecosystems. As I already told you, uncontrolled urbanization. You're using up too much land because our population is increasing at a rapid rate. Especially, I think our country has now become the world's leads the world in the population. So deforestation, okay, uncontrolled urbanization, deforestation. These will reduce the habitat space that is available for the birds and animals, okay, which are living in the forest. So they try to come into the urban land, okay, and changes in the food production patterns. You can see this has led to the emergence of E. coli, population getting displaced, the same thing. And I already discussed the problem of refugees, displaced persons, overcrowding is there. So you have air pollution because of that, as well as diarrheal and intestinal parasitic diseases spreading. And this is one example. See, actually in Venezuela, they started clearing a lot of forest area, and that resulted in an increase in this cane mouse population. And this mouse is believed to be a vector for this Guanarito virus. And this in turn led to an outbreak of the Venezuelan hemorrhagic fever. So that is an environment related factor. And then you have these wind changes occurring, isn't it? Even the rainfall is in India is determined by this El Nino phenomenon that also determines triggers of natural disaster as well as outbreaks of infections like malaria and cholera. And it has also possibly led to the increase in epidemics of this Rift Valley fever and Sin Nombre virus and all that. And another feature is building dams. We are trying to slow the, when you build dams, you try to collect more water, but at the same time, the water flow is slowed. So this changed the direction of in which the snails go. And snails are supposed to be the reservoirs for this. So it led to an emergence of this Schistosoma manzoni in Egypt. Okay. And it increased its occurrence in the Nile Delta. So that is another factor, an environmental factor. And heavy rains. Okay, global warming is said to increase the incidence of malaria, dengue, leishmaniasis, filariasis, okay, as well as fungal infections. And whenever there's a salinity decrease, okay, there is an increase in the toxic bacteria. And again, a lot of, when the vegetation increases, there's an increase in the number of rodents. And usually when there's heavy rains, what happens? There's water flooding. So that increases the breeding sites for mosquitoes. And you would have seen that in our country, then the incidence of dengue fever goes up after June, because after June we have heavy rains, especially in our state. Let's go on to the case studies. So I'll just, this was, the first one is COVID. So let's see the timeline of this COVID-19 disease. So in 2019, in December, so this was in 2019, they first diagnosed cases of pneumonia. The etiology was unknown, reported in Wuhan. Okay, and after that, the Chinese Center for Disease Control, they found out or diagnosed it as a new coronavirus from the admitted patients. And soon in January 9th, there was a first death reported due to COVID. And then after that, in 2020, January 13th, you see there was one case outside China. And then by that time, you know, in January 2020, okay, almost 282 cases. So right from 3 or 4 cases, it just jumped to 282 cases and six deaths. And during this time, the WHO kind of declares it as an outbreak of public health emergency of international concern. So that's what they did. And then again, there was a death in Philippines in February. And then now they renamed it as SARS-CoV-2, and the disease is COVID-19 disease. And then finally in March, WHO announced it as a pandemic. And then if you see by August, you had so many cases, okay, 21,294,000 cases. And by that time, 761,000 deaths. And after that, we all know how all of us were affected by COVID, okay. So how did this happen? So the first mysterious case of this pneumonia was detected in the city of Wuhan, okay, in China's Hubei province. And it was detected as a novel coronavirus, okay. And these wet markets, what you call it as wet market is because they are just selling live animals. Both there is illegal sale as well as a legal trade. And all the body secretions are going to be present there, okay. So because of that, you can see the people coming in contact with these body secretions of these animals, the blood of these animals, they may be infected, they may not be infected. So that is how it spreads. So what exactly happens is that this animal is from either bat or pangolin. Okay, this is the picture of the pangolin, and these are the bats in the market which are being sold. These two were only considered to be the main source of infection. Okay, so they are zoonotics because they are from animals. And these bats and pangolins were sold live or dead, okay, in these wet markets. And they sell the meat. And why we call it as a wet market is, as I already told you, all these body secretions are also there, body fluids, urine, feces and all there. Plus there is poor hygiene and infection control practices followed here. So because of that, this this virus is able to jump from the animal host to the human host. So the first thing is it has jumped the host from animal to the human. So it has broken the species barrier. And once it has entered into the human body, it has a unique feature to mutate rapidly. So because of that, it's able to adapt to the new human host. So once it comes to the human host, human to human transmission occurs by direct contact with these droplets, okay, released during conversation, coughing, sneezing and all that. So you know that this person is touching, got the infection from the animals, but he is the source of infection for the other people in the community or in the hospital, to other patients or to the healthcare workers. So that is how COVID started spreading. Then another case study is H1N1, where we all know that it was known as swine flu, isn't it? In 2009 when it emerged. But actually it is not only from the swine, okay? But what happens is the pigs can get infected. Why we call it as swine flu is that pigs, they can get infected from the human strain also, okay, as well as from the bird strain. So what happens is the pigs act as a mixing vessel in which influenza virus from different species can, multiply at the same time. And then what happens is that it can swap genes. I just want you to know that the influenza virus is an RNA virus. It is a mixovirus and it has got an 8-segmented genome. So the genes can undergo a reassortment, and then you can have mixing of genes between 2 strains or even 3 strains. And because of that only, you had a totally novel strain. It is known as the novel H1N1 or novel influenza virus. It has got genes from the avian, that is the bird, the human, as well as the pig strains of the virus. So actually it is known as a quadruple genetic reassortment, okay? And it shows that it has got a 30% of the North American Just a minute. Yeah, it has got a 30% origin from the pig, the North American swine. It's got a 34% gene origin is from the North American bird strain. And then you have the human influenza strain is 17%, and Eurasian swine strain is 17.5%. This Europe, that's because of export of these pigs to the American continent. So that is how you had a mixture of all the 4 strains and led to a totally novel strain. Okay, so that is another example of an emerging thing. The third story which I'm going to tell you is about Nipah virus. It is also a zoonotic re-emerging virus, because in 2018, what we had was a re-emergence. It was not an emergence because it was already in existence. But when did it first appear? It was seen in Malaysia and Singapore. See, again, here pigs are the kind of the reservoirs, okay, the mixing vessels, you can say. And this the Nipah virus belongs to the genus of Henipa, okay, and it's a paromyxovirus. And the reservoir for this virus is the fruit bat. Okay, it's also known as the flying fox. The genus is Pteropus. So these fruit bats, and these infected fruit bats will spread the disease to other animals or to human beings. Okay, so this is the story behind it. So in Malaysia, there were a lot of pig farms, okay, which were established in the areas where deforestation had been carried out, and there were a lot of fruit trees. And so they attracted the bats from the tropical forests. And these pigs now were which were grown in the farms, okay, they were exposed to the bat urine and feces because they were feeding on the fruits in these trees. And these initiated the infection in pigs, okay. The pigs were now exposed to the excretions as well as the secretions of these bats. And then between intensively reared between the pigs itself, it was spreading. And then finally through fomites, okay, items of clothing, equipment, boots, vehicles, the human beings got infected from the pigs. And then from human beings to other human beings, it spread via the hospital. And in 2018, there was an outbreak in Kerala. You'll know that only 18 cases were there. 17 people died because it's such a deadly virus. It causes both respiratory as well as encephalitis presentation kind of thing. And how we are infected is close contact with the infected animal, the body fluids such as saliva and urine. Okay. And once the infection spreads from animal to person, then it starts spreading from person to person. And the first spread from animal to a human being is only known as a spillover event. Okay. So this is the third case study we have seen. So we have seen about SARS, how it spreads. Okay. Then H1N1, we saw the story about the genetic reassortment, the quadruple genetic reassortment. Now we quickly look about Ebola. So Ebola virus is a severe hemorrhagic fever virus. You would have seen about it in movies. Even one Tamil movie you would have seen about in 2008. And it was named after the river Ebola in Congo, from where it was first identified. And just know that a lot of outbreaks have occurred in DRC, is Democratic Republic of Congo, as well as in Sudan. And each time it's always related to some episode where virus has been introduced from animal to a human being. Okay, so it's a direct spread from animal to human being. Usually it is hunters. The index cases are always hunters who get infected while handling the dead animals, their game, right? They kill the animal and then they take it as a trophy. So either gorillas and chimpanzees, the human beings, the hunters get infected. And then very fast that there will be an outbreak because it spreads very quickly between people. Mainly first the caregivers get affected. So in humans, how we get affected is multiple organ failure with internal and external hemorrhaging. So it's a very deadly virus. Okay. See, the case fatality is almost 90% in the Zaire type, and the Sudan type is almost 50%. Okay. Then a word on deliberately emerging infections. So we know what is bioterrorism. Deliberately we use some infectious agent, okay, by terrorist group or people who are not very happy about their country, and they create panic. Then they create mass casualties, okay, and destruction. Okay, they can affect, it can be targeted against human beings or against livestock. These agents, why they are called, they're very easy to mix in something, like mix it in water or something, or spread it like an aerosol, okay, and they're very difficult to detect it. And the ones with highest potential to be used in bioterrorism is Bacillus anthracis. This was the black dot which I showed you on the map. And this occurred soon after the 9/11 bombing episodes in 2001. And Apart from that, you have Clostridium botulinum, okay, Francisella, Yersinia, Variola, and viral hemorrhagic fevers can also be used for this. And these are emerging bacterial infections in immunocompromised people, non-typhoidal Salmonella, Campylobacter, Bartonella henselae, okay, Mycobacterium marinum, and Capnocytophaga. So how do we control these emerging infectious diseases? 4 main subheadings are there. So surveillance and response. I will tell you just a few points about this. Applied research, okay, infrastructure and training, prevention and control. That leads to all this. So in surveillance and response, so any unusual, unexpected, unexplained disease patterns, we have to have the ability to rapidly detect it, and you have to track it, exchange information in real time, okay, and immediately initiate effort, okay, and then this response should be taken globally. This can help to contain the transmission swiftly and very effectively. So something to that effect has been coordinated by the WHO, which is known as the GOARN, okay, Global Outbreak Alert and Response Network. And this one only combats all the international disease outbreaks. Okay, they do surveillance, they perform the relevant investigations, they initiate in early control measures and all that. Okay, and they implement behavior-related preventive measures, political and environmental measures. They also monitor and then evaluate all the effectiveness of our control and preventive measures. I told you about research. So where does research come into play? We all know rapid diagnosis. In COVID, we saw how many PCR kits were being introduced. So you just do research and bring out rapid diagnostic kits. New drug development, okay? New vaccines. Either it can be new vaccines or whatever is available. Try to decrease the dependency on the cold chain. You know that you have to transport the vaccines to the delivery place by means of a cold chain, right? Decrease the cost of the vaccine, develop new vaccines, new strategies for delivery of the vaccine. By like everything need not be an injection, something like an inhalation or ingestion. That's the meaning. And then improve the vector control, use proper safe insecticides or non-chemical organic strategies can be used. You know, right? Nowadays, some fish are being used to take care of the mosquito larva. So some kind of an organic strategy. Okay. And how do you improve your infrastructure to beat the emerging diseases? Improve the response capacity. That is the first thing. Improve the surveillance, improve the diagnostic capacity in your country, the communication systems, and rapid data analysis strategy also you should have. Okay. And you have to educate the public, okay, about the use of, like for COVID, you would have seen that everybody knew that they had to wear a mask and perform the hand hygiene. So that was the effectiveness of the public health education. Okay. And you have to also educate people on preventive measures based on the mode of spread. So suppose it is an insect borne, how to control the insect, how to repel the insect, like that. Decrease inappropriate use of drugs. Okay. When you have a viral infection, try not to take an antibiotic. Okay. Something similar to that. Then decrease the use of antimicrobials in your agricultural products and in your food products, and encourage vaccination. There are some people who are scared of vaccination. So I think you'll have to educate your people, okay, and tell them to take the vaccine wherever it is applicable. And public health people, how they can help is, they can help to establish the surveillance, increase their lab capacity, okay, to investigate new agent, develop plans for handling outbreaks, inform the physicians about the responsible antimicrobial prescription and use, educate the public, help in vector control, and that's how they promote the health. And they should also be always on the lookout. Always you should anticipate. See, the first thing in any game is anticipation. What the organism will do. Okay, in a game, what your opponent will do. So same way, right? And what is the role of microbiology? We are the crucial link. We have to correctly identify, and on time we have to report. Okay, or there's no use in reporting after 10 days for something which you have to give the report in 12 hours or 24 hours. Okay. And then we have to work on the antibiogram also. And in any case, we see something which is not an ordinary thing, which is uncommon, you have to immediately report it. And the modern generation of microbiologists, they should be familiar with all these identification modalities. Okay, for the newer generation technology also, they have to be familiar. And these are my references. Okay, you can go through the WHO guidelines, the CDC guidelines, as well as all the research articles that I have displayed on, along with the author names. And thank you for your patience.
Thank you, ma'am.
Thank you.
So, participants, do you have any queries? If you have any queries regarding this session, you can mention that in the chat box. I repeat again, participants, if you have any queries. Yeah, ma'am, here is a question for you. Doctor, what is your opinion when some people were suggesting investing as a replacement of COVID-19 vaccine during this pandemic?
Actually, during the 2020, that is the first wave and the early second wave, they were giving the ivermectin also because it was supposed to be an immunity booster. But right now, the studies have proved that it is not a replacement for the vaccine, and it's just like it has only a placebo effect.
And there's one more question from Sarah. That was an eye-opening and incredible. I'm sorry.
That's not a question. I think she has made a comment, and thank you, Sarah, for your comment. Thank you.
Participants, is there any other queries for ma'am? Ma'am, here is one more question. Doctor, how can we persuade people to take the vaccine?
Yeah, that's a good public health related question. See, actually, we only have to talk to them in such a way that they can accept it. Okay, like how can you persuade a child to take a medicine for the first time? What do? You tell about all the good effects of the vaccine. You tell about the ill effects of the vaccine. Then you allow them to. I mean, you can give examples. You can show them, right? Like if they take the vaccine, what are the main diseases that can be prevented? At least for the main illnesses, you can take the vaccine. Still, there are so many people. Hello, can you hear me? Yeah, still there are so many people who are so scared to take the SARS-CoV vaccine. And this is actually a very tough task. Thank you.
So any more questions from participant side? I hope all the participants are clear with your explanation, ma'am.
Thanks, Bhargavi.
I request all the participants to switch on their videos so that we can have a snap for the day 2. I repeat again, I request all the participants to switch on their videos to have a snap. Thank you, participants, for the cooperation with that.
Thank you, and have a nice day, all of you. Thank you.
Thank you, ma'am. Thank you for joining us today.
Yeah.
BioLEAGUES is a not-for-profit professional association which prominently promotes research and development. We at BioLEAGUES have brought a revolution in the field of worldwide conferences. The conferences organized by BioLEAGUES brings together the professional wizards and leaders who have explored all the avenues in the field of life sciences, medicine, and technology. BioLeagues conducts events worldwide which help in enhancing the skill set of the people from diverse industries and forms a common platform for eminent personalities, physicians, researchers, doctors, and academicians, business figures, and much more. BioLeagues conference encourages better comprehension about improvements and progressions over the world. We work with our motto of creating a better tomorrow by organizing conferences and creating a network which will help grow a better tomorrow with the help of advanced technology and achieve. Here is the short glimpse of our BioLeagues.
Provides a collaborative platform for researchers, scientists, and professionals to exchange knowledge, ideas, and innovations through its organizations IFERP and BioLeagues. IFERP is dedicated to advancing research and innovation in engineering, technology, and management, while BioLeagues is committed to advancing scientific research and innovation in various fields of medicine, life sciences, and technology. 5 major associations of BioLeagues: International Association of Oncology, International Society for Renal Research, Association of Pharmaceutical Research, Universal Society of Food and Nutrition, Society for Nursing Practices. BioLeagues activities provide a platform for researchers to showcase their work and engage in thought-provoking discussions on cutting-edge research. Our activities includes international conferences, webinars, workshops, faculty development programs, BioLeagues has successfully organized several national and international conference series. Our associated partners. At BioLeagues, we believe that collaboration and innovation are key to advancing scientific research and making a positive impact on the world. Join us and be a part of this exciting journey. TechnoRead is a leading professional association that is committed to advancing research and innovation across the globe. TechnoRead provides a collaborative platform for researchers, scientists, and professionals to exchange knowledge, ideas, and innovations through its organizations IFERP and BioLeagues. IFERP is dedicated to advancing research and innovation in engineering, technology, and management, while BioLeagues is committed to advancing scientific research and innovation in various fields of medicine, life sciences, and technology. 5 major associations of BioLeagues: International Association of Oncology, International Society for Renal Research, Association of Pharmaceutical Research, Universal Society of Food and Nutrition, Society for Nursing Practices. BioLeagues activities provide a platform for researchers to showcase their work and engage in thought-provoking discussions on cutting-edge research. Our activities includes international conferences, webinars, workshops, faculty development programs, BioLeagues has successfully organized several national and international conference series. Our associated partners. At At BioLEAGUES, we believe that collaboration and innovation are key to advancing scientific research and making a positive impact on the world. Join us and be a part of this exciting journey.
Here are some upcoming events of BioLeagues. Here is the conference which is going to happen in Association of Pharmaceutical Research, 10th World Conference on Pharmaceutical Science and Drug Manufacturing, scheduled on 6th and 7th July 2023, a 2-day conference happening virtually with the theme of an integrative and holistic approach towards pharma science. The scientific partner for the conference is BioLeagues and the research partner is United Innovators. So APR is delighted to welcome all the participants to join the conference as a presenter or as a listener. Now, the registration slots are open for the conference as a presenter as well as a listener. Early bird registration offer is going on. To avail that early bird registration offer, participants can register on or before 10th May 2023. People, if they are interested in giving participation and presentation, they can submit their abstracts by 15th May 2023 under the categories of applied pharmacology, clinical pharmacology, pharmacogenomics, nanotechnology in pharmaceutics, current therapeutic drugs, pharmacokinetics and dynamics, drug discovery and development, pharmacognosy, toxicological researchers, toxicology and its types, toxicogenomics, pharmaceutical biotechnology. To register, please scan the QR code displayed in the poster. And if you have any queries regarding this, you can contact us on the numbers displayed in the poster.
Hello. Hello. Hello, can you hear me?
Yes, we can hear you.
Yeah. I wanted to know, I couldn't figure out where are the breakout rooms here.
Breakout rooms are gonna open in few minutes. Once it is open, we'll assist you in joining.
Okay,
in the
so it's
breakout
not
room.
yet open. Yeah.
Not yet open.
Thank you. Thank you.
Yeah, thank you. Here is one of our upcoming conferences happening in Society for Nursing Practices, 6th International Conference on Nursing Science and Healthcare. in association with BioLeagues, powered by Technored, and research partner is United Innovators. This conference is going to happen on 16th and 17th June 2023 in Jakarta, Indonesia. This is a hybrid conference happening with the theme of Nurses Together for Global Health Advancements. So the early bird registration for this conference is also going on. Students who are interested in joining for the conference can avail the early bird registration offer till 30 April 2023. The conference topics are nurse career and education, nursing research and evidence-based practice, nursing in emergency medicine, patient safety and healthcare, advanced practice, registered nurse, nursing safety and legal nursing, future of nursing education, medical surgical nursing, child health nursing, mental health nursing, community health nursing, pandemic crisis management, recent technologies in nursing, shortage of nurse in pandemics, emergency preparedness, nursing research and patient-centered care, obstetrics and gynecology. These are the topics for the conference. Participants can submit their abstracts on or before 10th May 2023. And last date for the online registration is 20 May 2023. If participants are having any queries regarding this conference, you can contact our team at any time, and the numbers are displayed below in the poster here. Here is an announcement for the participants. The breakout rooms have been opened for the conference. People who are supposed to be there in Hall A, please start joining for Hall A. And participants who are having their presentations in main hall are requested to stay back in the main hall. I repeat again, breakout rooms for the conference have been opened. Participants who are supposed to have their presentations in the main hall are requested to stay back in the main hall. And participants who are having their presentations in Hall A are requested to join in Hall A. Hello, participants, can you hear me? Am I audible to you? Yeah, thank you for the response. So now we are going to start the technical sessions. I would like to welcome and introduce session chairs for the today. Dr. Mukta Sharma, Professor and Deputy Dean, Microbiology, IIMT, University, India. Dr. Srijit Das, Research Scientist, University of Iowa, United States of America. Session chairs, with your permission, can we start the technical session?
Sure.
Yeah, thank you. So for the today, the first participant is unavailable. He might be joining in a while. So we are going with the second presentation by Shivangi Srivatsava, oral presentation from Sister Niveta University, Kolkata, on topic review on CAR T-cell oncoimmunotherapy. Shivangi Srivatsava, I hope you are available online and you can give your presentation.
Good morning. Am I audible?
Yes, you are audible. You can start sharing your screen.
Is my screen visible?
Yes, your screen is visible.
Good morning, everyone. I'm here to present on CAR T cell therapy. My name is Shivangi Srivastava. I'm a student from Sister Niveta University. And I've done this review under Dr. Atri Ghosh ma'am. Now we are aware how cancer has tormented us throughout our life. Of human life. Cancer is a disease in which some of the body's cells grow uncontrollably, often forming a mass of tumor, spreading to other parts of the body and destroying body tissue. Usually, the human immune system has a defense mechanism or soldier T cells to invade foreign or damaged cells. Including oncocells and limit damage to healthy cells in case of infected and other diseases. These T cells have protein receptors that bind to the antigens of the foreign cells and release some toxins and kill the cells. Now, for centuries, cancer treatment has been surgery, chemotherapy, and radiotherapy. But some new treatments have arisen in the recent decade. Like immunotherapy that uses immune systems, body's defense mechanism against cancer by using immune checkpoints, blockage, adoptive cell, and transfer vaccines. The CAR T cell therapy is a form of cancer therapy that involves genetically modifying a patient's own immune cells called cancer T cells to recognize and attack cancer cells. The therapy has been successful in treating certain types of blood cancers, such as acute lymphoblastic leukemia and diffuse large B cell lymphoma that have not responded to traditional cancer methods. Now, that we are known that normal T cells were able to remove the foreign cells, but then what was the need of this cancer T cell? The cancer had acquired mechanisms to escape the normal immune defense by disguising themselves as healthy cells or sprouting multiple antigens and hinder the attack and finding ways to turn off the immune system mechanism. Now, how are they produced? They were produced by using a modified lentivirus that was did not pose any other harm. use the DNA to sprout chimeric antigen receptors on the cell along with the normal receptors. The CAR T cell receptors latched onto the tumor antigen via CD19 receptor and released cytokines and toxins and destroyed the cells. Moreover, they remain in the body and reactivates if the cancer returns even after years. The CAR T cell therapy proved to be successful in cancer cases like lymphoma and blood cancer. Now, how was this therapy cell performed? First, the T cells were extracted and genetically engineered. And cloning of the T cells was taken place and they were reintroduced into the patient. Here the cells, T cells, normal blood is taken out and T cells are isolated and then genetically modified and multiplied and again injected into the patient's body. This diagram shows the location where they take place. The structure of the CAR T cell. They had an ectodomain that attaches to the tumor cell, this part, and the transmembrane, this part that provides stability. So this is the tumor cell and this is the ectodomain. And this is the transmembrane. And the endodomain had functional ends on the receptor. The advantages, it had proved a high efficacy and had a targeted approach and had the potential for long-term remission. The limitations, there were quite a few limitations. The potential for severe side effects like CRS, neurotoxicity, and that could be life-threatening too. They had limited applicability as till now it has only proved to be beneficial for blood cancer and not effective for all other cancers. They are still under study. And also it had a high cost. The precise cause for this, neurological side effects is not yet known and they are under study. So it proved to be a limitation. The future prospects include expanding the use of CAR T cell therapy to other types of cell, the development of more targeted CAR T cell therapies, combining CAR T cell therapy with other cancer treatments, improving safety profile of the CAR T cell therapy, and developing off-shelf CAR T cell therapy, meaning now the blood was taken from the patient. They are trying to take blood from other donors and then trying to incorporate those T cells into the body. The clinical trials proved very beneficial. Then this is the global CAR T cell therapy market overview. Earlier, it will rise by 38.7. It is estimated, and it has been used in many locations till now. Now, I would like to conclude by saying overall CAR T cell therapy represents a promising approach to cancer treatment, and has shown successful clinical trials, but there are still challenges that need to be addressed. Ongoing research and development will be critical to further refining and expanding the use of this therapy. There are ongoing trials and new trials in the solid tumors, blood cancers under study, like multiple myeloma and HER2 breast cancer. Then we also know that now CAR T cell therapy has made the human body into a cancer fighter. There are new types of CAR T cell therapies under research too, like light switchable CAR T cell, and the FDA approved the CAR T cell therapies also. I would like to acknowledge, I would like to convey my heartfelt gratitude to my HOD ma'am, Dr. Atri Ghosh ma'am, and I would like to thank BioLEAGUES also. Thank you. These are my few references, and I'm open to any questions. Thank you for your attention.
Thank you, Sivangi. Session chairs, any questions for her?
Uh, Hello, am I audible?
Yes, yes.
Shivangi, very good presentation. I have some question. Why we need CAR T cell?
Yes. Actually, these, I would like to refer to my slide. We all know that earlier these were used like radiotherapy, and they're still used, but uh, they are the radiotherapy and the chemotherapy are very painful, right? But the CAR T cell therapies are not as painful, and the cancer may return to. But over here in CAR T cell therapy, the cancer, even after returning, the cancer cells can be again, these CAR T cells remain in the body and they get reactivated, and they prove to remove the cancer cells.
How these cell can work?
Sorry.
How CAR T cell can work?
The CAR T cell releases certain cytokinins in when they attach to the tumor cells. They have now they earlier they use normal T cells had certain receptors, but the CAR T cell receptors have a chimeric antigen receptor attached to it. Then these chimeric antigen receptor attached to the tumor's antigen and release certain types of toxic cytotoxins and kill the cell.
Shivangi, it means you are saying that these cells are the equivalent to giving patient a living drug.
Yeah.
Am I right?
Yeah.
So, is there any side effect of these
Yeah,
CAR T
there
cells?
are many side effects like CRS. I would like to refer to my slide. Yeah.
CRS is cytokine release syndrome.
Yeah, and ICANS. There are many neurotoxicity syndromes also. In CRS, there are fatigue and headaches and also occur, and they can be fatal too.
What exactly happened in this CRS, in this syndrome? What exactly happened neurologically?
Actually, I haven't studied that much.
I think cytokines multiply. Cytokines multiply when CAR T cells enter into the body.
Yeah.
Am I?
Yes.
I think it should be. Thank you, Shivangi.
Thank you, ma'am.
Hi, Shivangi. Good presentation. I don't want to ask you more questions, but I just wanted to tell you that so when you use any figure or any cartoon from any either website or any paper, so you should always refer that, okay? Because I've seen in some of your some of the figures and cartoons that you used, I don't see the references, like meaning from which paper or which book or wherever you got it from, okay?
Okay.
But good presentation. Thank you.
Thank you. Should I stop presenting?
Yes, you can.
Okay,
Thank you. Thank you for presenting, Shivangi.
Thank you.
So we are moving with the next presentation by Nithyamala I from National Institute of Siddha, going to give her oral presentation on the topic a review on antimicrobial activity of Siddha herbo mineral formulations. Nithyamala, are you there available for the presentation? Nithyamala, I hope you are available for the presentation. Nithyamala, you can start sharing your screen. Dr. Nityamala, if you have any technical issues, you can let us know. We'll be helping you in figuring that out. Dr. Nityamala, can you hear me? Sorry, you are not audible. I hope you are having some technical issues. You can clear that. Meanwhile, we'll be going with the other presentation. So we are going with the other presentation by.
Can you hear me now, ma'am?
Yes, ma'am, we can hear you.
Can you see the PPT, ma'am?
Yes, we can see that.
Shall I present, ma'am?
Yeah, you can go with that.
Okay. I am Dr. Nityamala, Medical Officer in National Institute of Siddha. I am going to present on the topic a review on antimicrobial activity of Siddha herbal mineral formulations. Introduction: Indian system of traditional medicine like Siddha pioneers the art of healing from ancient time, as it is systematically documented and well established. Practice guidelines framed by the traditional healers, otherwise called as Siddhas. Siddha text prescribed several lakhs of medications and formulations that are indicated for various diseases. Siddha raw drugs are classified into three types: Mooligai herbs, Dhatu metals and minerals, and Jeevam animal products. There are plenty of herbo-mineral formulations formulated by the Siddhas. This presentation reviews about the antimicrobial activity of some herbo-mineral formulations used in the Siddha system of medicine. The antimicrobial activity of herbo-mineral formulations in Siddha. First, Tazhambu Matrai. Tazhambu Matrai was obtained from the ICCAM pharmacy, and the stock solution was prepared by dissolving the dried extract in DSMO at various concentrations.
Yeah.
Hello. Ampicillin was prepared with sterile water at a concentration of 10 milligram per ml. The antibacterial and antifungal assay was carried out. The microbial activity evaluation was carried out by couplet method. It's a review article. I have selected some research studies for this. The organisms used were E. coli, Klebsiella pneumoniae, Proteus vulgaris, Salmonella typhimurium, Pseudomonas aeruginosa, Bacillus cereus, Staphylococcus aureus, and the fungal strain Candida albicans was tested. The test drug was effective against Pseudomonas aeruginosa at a concentration of 0.09 mg per ml and produced an inhibitory zone of 27 mm at 50 milligram per ml. At concentration of 0.78 milligram per ml, the drug was effective against Proteus vulgaris, and at 50 mg per ml concentration produced an inhibitory zone of 15 mm against the same. Maximum zone of inhibition at a drug concentration of 50 milligram per ml against Bacillus cereus, Staphylococcus aureus, and Salmonella typhimurium was measured to be 15 mm, 17 mm, and 19 mm respectively. The drug concentration to be effective against Candida albicans was 0.39 milligram per ml, and the zone of inhibition produced against the fungal strain was 19 mm at a concentration of 50 mg per ml. The Tazhambu Matrai was found to be effective against two bacterial strains, E. coli and Klebsiella pneumoniae. Next, second drug is Kanagalinga Meligu. The study was carried out at the Satyabama University, Chennai. The pathogens studied were E. coli, Klebsiella pneumoniae, Proteus mirabilis, Staphylococcus aureus, Pseudomonas aeruginosa, Shigella soni, Aeromonas hydrophila, Salmonella typhimurium, Vibrio cholerae, etc. The Siddha formulation Kanakalinga Medical showed moderate broad spectrum antimicrobial activity against gram-negative and gram-positive organisms. The organism E. coli showed a higher zone of susceptibility, approximately 70% against standard positive control ciprofloxacin. The test drug was approximately 50% as sensitive as that of ciprofloxacin against other organisms. It had very good sensitivity of around 88% as that of clotrimazole and beclomethasone against Candida albicans a fungus. Third drug is Dasalavana dravagam. So Dasalavana dravagam extract was prepared and the microorganisms studied were Proteus vulgaris, Enterobacter aerogens, E. coli, Pseudomonas aeruginosa, Klebsiella pneumonia, Salmonella typhimurium, Staphylococcus aureus and Bacillus subtilis. The antimicrobial activity was carried out by the couplet antibacterial susceptibility test. The effect of Dasalavana dravagam exhibited significant zone of inhibition in 4 organisms out of the 8 selected microbial screening, in which the maximum inhibition zone diameter of about 26 mm against Staphylococcus aureus at the volume of 60 microL per well and the minimum IZT of about 0.09 mm at the volume of 10 microliter per well against the same. Second best was exhibited by Klebsiella pneumoniae at the volume ranging from 15 to 60 microliter per well. Dasalavana dravagam is highly sensitive against Klebsiella pneumonia, Salmonella typhimurium, Staphylococcus aureus, Bacillus subtilis, indicating its promising antimicrobial potency against selective gram-positive and gram-negative organism. Further, the test drug fails to prove its antimicrobial activity against Proteus vulgaris, Enterobacter aerogens, Escherichia coli, and Pseudomonas aeruginosa. In the next study, Gauri Chindamani, Sivana Ramadam, Purana Chandrodayam, Talaga Parpam, Pavala Parpam, and Vasanta Kusumagara Mastri were studied. It was carried out in the Anna University MIT campus in Chennai. The pathogens were Neisseria mucosa, Klebsiella pneumonia, Streptococcus pneumonia, Staphylococcus aureus, Pseudomonas aeruginosa, and Aspergillus niger. Agar well diffusion assay was carried out. The sample Gauri Chindamani showed activity against gram-negative organisms. Neisseria mucosa, Klebsiella pneumonia, and Pseudomonas aeruginosa. The formulation Sivanaramirtham and Pavalaperpum showed broad-spectral antibacterial antibiotic activity against all the tested gram-positive and gram-negative organisms, of which the drug Sivanaramirtham showed higher zone of inhibition than the positive control against the pathogens N. mucosa, Klebsiella pneumonia, Staphylococcus aureus, and Pseudomonas aeruginosa. The sample Vasanta Kusumagara Mastri showed specific antibacterial activity against Staphylococcus aureus and Pseudomonas aeruginosa. The samples Purna Chandrodayam and Talaga Parpam did not show any antimicrobial effects against any of the tested pathogens. In the next study, the Nandikal Parpam, Rasagandhi Medigu, and Karkaraichi Matri, 3 herbomineral formulations were studied. The method adopted for this is Kirby-Boker disk diffusion method. In this study, the extract was prepared and the culture was also prepared. Antimicrobial sensitive testing was done using Kirby-Boker disk diffusion method. From the results, the Karkaraichi Matre showed significant increasing zones of inhibition towards E. coli with increasing volume of the extract. This evidences that Karkaraichi Matre acts considerably against E. coli. At 20% concentration of the extracts, Nandikal Parpam showed significant activity against Staphylococcus aureus. Rasagandhi Medigu showed activity against Staphylococcus aureus and Pseudomonas. Karkaraichi Matre showed activity against all the tested uropathogens. Summary of the findings of the review article. Tarambu Matre showed activity against the following organisms: Pseudomonas aeruginosa, Proteus vulgaris, Candida albicans, Bacillus aureus, Staphylococcus aureus, and Salmonella typhimurium. Kanakalinga Medigu was effective against E. coli and Candida albicans. Dasalavana dravagam was effective against Staphylococcus aureus, Klebsiella pneumonia, Bacillus subtilis, Salmonella typhimurium. Gauri Chindamani is was effective against the gram-negative organisms, Neisseria mucosa, Klebsiella pneumonia, and Pseudomonas aeruginosa. Shivana Amritam was effective against Neisseria mucosa, Klebsiella pneumonia, Streptococcus pneumonia, Staphylococcus aureus, Pseudomonas aeruginosa, and Aspergillus niger. Purna Chandrodayam did not show any antimicrobial effects. Similarly, the Talaga Parpam, which is a herbomineral formulation, did not show any antimicrobial effect. The Pavala Parpam was effective against Neisseria mucosa, Klebsiella pneumonia, Streptococcus pneumonia, Staphylococcus aureus, Pseudomonas aeruginosa, and Aspergillus niger. Vasanta Kusumaka Mate was active against Staphylococcus aureus and Pseudomonas aeruginosa. Nandikal Parpam was active against more preferable than herbal drugs. In the above studies, Kanagalinga Medaku, when administered for gynecological problems, can have an additional advantage of antimicrobial efficacy, which would be useful in the treatment of pelvic inflammatory diseases also. Karkaraichi Matre showed activity against all the tested uropathogens, so that it can be used in treating urinary tract infections. The formulations Sivanaramatham and Pavalapartham showed broad spectral antibiotic activity against all tested gram-positive and gram-negative organisms, of which drug Sivanaramatham showed higher zone of inhibition. Thus, the Siddha herbomineral formulations can be used for various antimicrobial activities in the clinical practice for treating various diseases. That's the conclusion, ma'am. Thank you. See, these are the references of the articles.
Thank you, Dr. Nityamala. Session chairs, any questions for her?
Hello, Dr. Nityamala.
Yes, ma'am.
Very good presentation.
Thank you.
I have a question.
Yes,
How you have prepared this formulation? You have talking about the Siddha.
Yes, ma'am.
How you have prepared this formulation?
Actually, we are getting it from the GMP standard companies, ma'am.
Okay.
In corps like that, we have some GMP certified companies to buy the drugs. From there, we are getting, ma'am.
Okay. So you have not formulated this in your lab?
No, ma'am. It's not in our lab, ma'am. It is it's bought from INCOPS.
Okay. You have mentioned the cup plate method in your study. So can you explain this method?
Ma'am, that I don't know that much about that method, ma'am. It's only.
And you have, along with this cup plate method, you have also mentioned the agar-well diffusion method. So what is the difference between these two methods?
Basically, I'm a Siddha doctor. I don't know that much about the methods, ma'am. I just have.
But you have mentioned in your study, na? So that's why you should know about these methods. Okay. Have you isolated these microorganisms? You have mentioned a lot of bacteria, E. coli, Staphylococcus and all. Have you isolated these microorganisms?
It was all done in some labs, ma'am. We don't have that facility in our institute.
You mean you have procured?
We are outsourcing the.
You mean you have procured from any other lab?
Yes, ma'am. Yes, ma'am.
And you have also mentioned that these formulation is effective against the UTI causing bacteria. You have mentioned in your study. So how can you conclude that this formulation is effective against the UTI causing bacteria?
The uropathogens were studied in the article, ma'am, so that we can conclude that it can be.
But you have not isolated these bacteria from the urine sample. If you have not isolated from the urine sample, then you cannot conclude that it is effective against the UTI causing bacteria.
Okay, ma'am.
This E. coli may be from any other source.
Oh, okay, ma'am. Now only I can understand.
You can conclude only if you have isolated from the uropathogens, right?
Okay.
Okay, otherwise your presentation is very good.
Thank you.
Thank you so much. Thank you so much.
Thank you.
Srijit sir, any questions from your end?
No, thank you. Good
Okay.
presentation. Thank you.
Thank you, sir. So with that note, we are going with the other presentation by Karpagavali from National Institute of Siddha for oral presentation on the topic, a review on antimicrobial activity of herbs used in Siddha system of medicine. Karpagavali, I hope you are available for the presentation. I hope she's having some technical issues. We'll be moving to the presentation of Arpita Jaha, oral presentation from Central University of Jharkhand, Ranchi, on the topic, the expression or polymorphisms of TLR 4/9, gingival inflammation or recession or bacterial infection can predict oral cancer incidence among the Jharkhand population. Arpita, I hope you are available for the presentation and you can
Yes.
start sharing your screen.
Is it visible now?
Yes, your screen is visible and you are also audible. You can go with the presentation.
Okay. A very good morning to one and all present over here. I, Arpita Jha, research scholar from Central University of Jharkhand, will be discussing my topic entitled The Expression or Polymorphisms of TLR4 and 9, Gingival Inflammations or Recession or Bacterial Infection can predict oral cancer incidence among the Jharkhand population. So to start with, we should know what is oral cancer and how is it related to immune system. So oral cancer is one of the most lethal diseases worldwide, and it causes around 40% of oral cancer incidences in India and ranks as 6th most common cancer in the world. There are many significant factors of oral cancer causes, such as drinking and smoking, chewing betel quid and betel nut, and human papillomavirus infections. But there are certain other factors that are given less importance but are involved in the causing of oral cancer. The human oral cavity is inhabited by around 750 different taxa of bacteria, and the immune system plays an important role in fighting against the infection caused by these bacteria. So the immune system consists of innate immunity and acquired immunity, and I'll be discussing about the innate immunity. So the innate immunity is also known as the first line of defense and is composed of granulocytes, basophils, eosinophils, macrophages, and dendritic cells, and also toll-like receptors, which helps in initiation of innate immunity. So toll-like receptors, also known as TLRs, are present on the membrane of different cells. So I have studied TLR4 and TLR9. The TLR4 is present on the membrane of epithelial cells, and it recognizes lipopolysaccharides, glycoinositol phospholipids, enveloped glycoproteins, and host-derived heat shock proteins. Whereas TLR9 is present on the membrane of endosomes, and it recognizes unmethylated CpG DNA and mitochondrial DNA. And once these recognition occurs, they help in the activation of certain transcription factors, that is IRF1, NF-kappa B, and many others, that ultimately leads into the activation of certain cytokines that helps in fighting against this infection and killing the foreign pathogens. So based on these literature surveys, the main aim of the study was to understand TLR4 and 9 expression, polymorphism, that is single nucleotide polymorphism, and their association with bacterial infection, gingival inflammation or recession, and OSCC, that is oral squamous cell carcinoma or oral cancer risk in Jharkhand population. So these are the different materials and methodologies used for fulfilling my objectives. So the first of all, 3 different types of samples were collected, that is post-operated oral cancer tissue samples, oral swab, and blood. After the approval of Institutional Human Ethical Committee of Central University of Jharkhand, then DNA RNA was isolated and different types of PCRs were used, and different types of statistical tools for the study of chi-square analysis, Hardy-Weinberg equilibrium is genotype, allele frequency, and haplotype analysis. So first of all, I wanted to study the expression of TLR4 and TLR9 in the oral cancer patients of Jharkhand. So here AN represents the normal samples and the T represents the post-operated oral cancer tissues. So I have observed that the mRNA as well as the protein expression of TLR4 and TLR9 was highly significant in oral tissue samples. And for the study of single nucleotide polymorphism through different literature surveys, I have selected different SNP IDs. And for TLR4, the SNP IDs over here are represented as N1, 2, 3, and 4. And for TLR9, the SNP IDs are represented as N5, 6, 7, and 8. So my first question was whether the TLR4 and TLR9 single nucleotide polymorphism is associated with oral cancer or OSCC incidences or not. So for this study, after the collection of diseased samples, that is oral cancer cases, tissue samples, and for the control, here the oral swab was collected, and for after that the DNA was isolated, and using allele-specific PCR, the genotype of each sample was determined, and after that the chi-square test was performed. And here I have mentioned only the significant p-values. So the significant p-values over here demonstrates that the specific SNPs of specific genotypes are associated with high risk of oral cancer. Then I wanted to study whether the single nucleotide polymorphism of TLR4 and TLR9 is associated with gingival inflammation and gingival recession and OSCC or not. So for this study, after knowing the genotype, I have categorized my sample into the diseased and the control. The control were same, which were non-cancerous and healthy and without any kind of infection, whereas the diseased were like oral cancer samples along with the infection of Porphyromonas gingivalis, Fusobacterium nucleatum, Treponema denticola, and this was the infection was identified using the 16S RNA PCR. So after the identification of the infections with one or more than one infection, it was categorized as diseased with oral cancer. And after this, again, the chi-square test was performed, and the significant p-values represented over here shows that specific SNP IDs along with the specific genotypes are associated with gingival inflammation and recession along with oral cancer. So based on these data, the haplotype analysis can be determined. The haplotype can be defined as a set of DNA variations such as single nucleotide polymorphism over here that are adjacent to one another and present at the same locus that tends to be inherited together. So based on the using a software, HaploView, I have found the haplotypes that provide risk to oral cancer and the haplotypes that provide protection against oral cancer. So the p-values over here represents that these haplotypes or specific SNPs, that is for TLR4 and 9, are causing oral cancer risk, and these haplotypes provide protection against oral cancer. Then after that, I have checked the expression of TLR4 and TLR9 protein and mRNA in the specific genotypes, and it was also found that the expression are significant with specific genotypes. So coming to the conclusion, I would like to conclude that TLR4 and TLR9 genotypes supports gingival inflammation and oral tumor progression, and the SNPs, that is SNP of TLR4 and TLR9 gene might be responsible for protein stability and overexpression of TLR4 and TLR9, and this overexpression leads to hyperactivation of certain transcription factors, that is IRF1, IRF3 and 7, NF-kappa B and AP1, which ultimately leads to the activation of inflammatory cytokines, which helps in the initiation of oral cancer progression as well as gingival inflammation and recession. And based on the statistics which I have discussed, suggest that some of these genotypes, alleles, and haplotypes could be used to predict oral cancer cases. And these single nucleotide polymorphisms of TLR4 and TLR9 statistics, along with the infections of certain bacteria that I have mentioned, can be applied in clinics as biomarker for disease prediction and risk assessment of oral cancer. Finally, I would like to thank my PhD supervisor, Dr. Rajikishore Mishra sir, for helping and guiding me in fulfilling these objectives, my lab members, Institutional Human Ethical Committee of Central University of Jharkhand, Department of Biotechnology and Indian Council of Medical Research for fellowship and funding, and different doctors of Ranchi who helped me in providing different samples and fulfilling this research. Thank you.
Thank you for your presentation, Arpita. Session chairs, any questions for her?
Hello, Arpita.
Hello, ma'am.
Very good presentation.
Thank you, ma'am.
I have only one question. Is there any relation of dental plaque or bacterial infection to TLR?
Ma'am, TLR are basically associated with different types of infections, which helps in the activation of innate immune system. So I think there must be some association with the plagues and different types of bacterial infection. But I have studied only these 3 types of bacterial infections, so I'm not very much sure about those infections. But I think this must be associated.
Yes, right. It is associated.
Yes.
Excellent presentation. Thank you so much.
Hi, Arpita. Can you hear me?
Yes, sir.
Yeah, very good presentation. So I was just wondering, like, what stage of these cancers, the OSCC samples, did you study?
So these are just the primary stage oral cancer samples, like non-treated with any kind of chemotherapy or non-FNAC positive also, like not invaded and not chemotherapy treated or not any kind of treatment. It's just the post-treatment or the, you can say that, biopsy samples I have chosen,
Okay.
which were confirmed cases of oral cancer.
Okay. And my last question is, do you know, like, is there any literature that shows this TLR4/9, these SNPs are also implicated in other types of cancers? Or is
Yeah,
it?
sure, sir. Yeah, sure, sir. As you can see, I have used one of my references. In the second references, it is associated with the cervical cancer also. And this is one of the paper which have been published by our lab members itself.
Okay. Okay. Thank you.
Okay.
Thank you.
Thank you,
Very
sir.
good presentation. Thank you.
Thank you, Arpita. Moving on to the other presentation. The other presentation is by Pradeep Reddy Anam, oral presentation on the topic susceptibility of Candida species to azoles and ERG11 gene mutation detection in azole resistant Candida albicans isolates. Pradeep Reddy, I hope you are available and you can go with your presentation.
Yes, ma'am. Can you see my slides?
Yeah, we can see you.
Good morning, everyone. I am Pradeep Reddy, PhD scholar at Bareilly International University. I have been studying this research question where whether what kind of mutations are present in ERG11 gene, which produces ergosterol. This ergosterol is the target for many of the antifungal drugs. So when there are any mutations in this ERG11 gene, the target receptors would be changed and the drug won't be acting as supposed to. So the incidence of Candida infections has risen to an alarming level in the past several decades, which is causing high mortality and morbidity. In the olden days, candidiasis was a very rare occurrence. Now it has become common for the bedridden and critically ill patients to become infected with Candida infections, despite the improvements of the medical care. So it can be said that the elimination of normal microbial flora of the gut by prolonged or use of broad spectrum antibiotics facilitate favorable conditions for Candida to overgrow and invade the anatomical barriers, which results in penetration into the deeper tissues and vasculatures. Although the epidemiological surveillance data suggests that Candida albicans is the predominant causative organism, single center studies have revealed a different picture. So that's the reason why I have taken up this study. Candida albicans is not the predominant species everywhere. It differs from place to place. In my study also, I found that non-albicans Candida are more predominant, more common in hospital settings than Candida albicans.
Sorry to interrupt
hi,
you, Pradeep. Your slides are not moving. We cannot see your slides moving.
Sorry, I did not move the slide actually.
Okay.
Can you make it full screen? It will be easier.
You can display it in the playing mode.
Can you see this now?
Yeah, we can see the screen of aims and objectives,
Yeah.
but it is not completely displayed. It's not in full screen.
This is in presentation mode and full screen.
We are sorry, we cannot see that. Okay, anyhow, continue the presentation then.
So now the aim and objectives are to find out the susceptibility or resistance of candida and non-albicans candida to 3 triazole drugs, fluconazole, voriconazole, and itraconazole. So we have collected the samples from patients, then identified the species, and antifungal susceptibility testing was done. Then the species of Candida albicans, which are resistant to all the 3 drugs, those are selected and they are subjected to gene sequencing. That is the ERG11 gene point mutations. We aim to study the changes in the gene. So this is the way materials and methods. Initially, the resistant azole resistant Candida albicans were selected. Then DNA extraction is done. ERG11 gene amplification is done by PCR. Then the quality and quantity is checked by gel electrophoresis and sequencing is done. So a total of 200 clinical isolates of Candida were isolated. Of these, 116 isolates were obtained from respiratory samples, which constitutes almost 58%. And from the urine, 34 samples, which is 17%. Then high vaginal swabs, 22 samples, that is 11%. So of all these, we found that 10 Candida albicans were resistant to all the 3 drugs, that is fluconazole, voriconazole, and itraconazole. So now Candida albicans, total of 43 were collected, isolated. So of these, 28 are sensitive to fluconazole, 33 samples are sensitive to voriconazole, and 26 to itraconazole. Only 10 are resistant to all the 3. So after performing the sequencing studies, we found a total of 83 mutations in all the 10 samples. Of these, 29 are unique mutations. 29 unique mutations were seen. Of these 29, 20 in the 20 mutations, amino acids were substituted, and in 9 mutations, there was no substitution of amino acids. A large number of clinical strains exhibited the same pattern of amino acid substitutions, that is the aspartate to glutamate substitution at position 116 and glutamate to aspartate substitution at position 266 and glycine to serine substitution at 464. These were the most common mutations occurred. They occurred 6 times in the 10 samples.
Hi, Pradeep.
Yes.
Sorry to interrupt you, but I'm still seeing your aims and objective slides. I don't know if it is my computer or.
I've been changing the slides, sir.
Okay. Anyway, go ahead. I mean.
So the summary is to conclude, this study highlights that there is a very high diversity and frequency of amino acid substitutions in ERG11 gene, which is the primary target of azole antifungal drugs. So this genetic polymorphism should be taken into consideration in the rationale of design of new antifungal compounds targeting this lanosterol 14 alpha demethylase, which is a product of ERG11 gene. So that's it, sir. Hello. Hello.
Hello, Pradeep.
Yes, ma'am. I have finished the presentation.
Nice presentation. I have a question. What is the mechanism of resistance to azole in Candida?
There are multiple mechanisms of azole resistance, ma'am. One is by excessive production of this ergosterol, and
Okay.
another method is by proton pumps, which prevent the drug entering into the cell in optimum
Okay.
concentration.
Okay. So, is there any solution of this problem? But the drug resistance is a very common problem nowadays.
Yes.
So, is there any solution?
It is yet to be found, madam, because the mutations are so varied and newer mutations are being seen. In this just 10 samples, we found around 58 mutations. So all in the same gene, which is the target for this enzyme, these drugs.
Hello, am I audible?
Yes, ma'am.
Okay, fine. Okay, thank you, Pradeep.
Thank you.
Hi, Pradeep.
Yes, sir.
I have a quick question. So, why did you pick this ERG11 gene? Meaning, are there other genes that's also known to be associated with azole resistance, or like?
Actually, there are other genes like MDR1 and CDR genes. This was selected because these azoles are the first choice of drugs to be given to any patients, and these are the safest of all the drugs. So that's the reason I selected this.
Okay. Thank you.
Thank you, Pradeep. We are going to the other presentation by Selvin, oral presentation on the topic exploring the limitless possibilities of machine learning and AI in medical microbiology, new developments and applications. Selvin, I hope you are available for your presentation.
Hello, ma'am. Am I audible?
Yes, you are audible. You can start sharing your screen.
Good morning, madam. My name is Selvin Barreto, and I work as a freelancer, medical microbiologist, medical laboratory technician. And my topic for today's presentation is exploring the limitless possibilities and learning of the medical microbiology, AI, in very new developments, and along with the applications, and finding out the different applications which can be done using AI and machine learning. So, say for example, you have a illness of any kind. Most of the experienced doctors who are there in the hospital or wherever you are admitted are unable to find out whether what is the infection that you're going through. All the treatments, all the tests you have done in your whole during the whole timeline is not working out in figuring out what is the infection that you have gone through, and all giving any personalized medication is a bit irrelevant to you, and time is very essential. In this scenario, AI, that is artificial intelligence, or a machine learning, will be helpful. And as we see in the 2023 era right now, we have different AI websites. We have the AI's softwares which are helping the medical healthcare in finding out what is the core infection, or what is the core causes for a particular infection. What is the microbes that is causing you such a pain and such a drastic uh, infection, and will help us in finding out what is the best treatment and given in the course due of time, and how it can be helpful in you in becoming a better person. So yes, so I'll be speaking on these 4 topics, that is artificial intelligence, then coming to the machine learning, uh, artificial intelligence microwave, the challenges and the limitations that we are going through right now, and along with that, the current applications. So now coming to artificial intelligence, what is it? I'm going to speak on 10 topics. So we'll go short by short. So the first topic is integration of machine learning, and first one is the basics one. Let's go with the basics. Over here, you see a picture of a cat. Let's just not look into it much properly. So basically, machine learning is like machine learning is a broad topic on its own, and machine learning is like a one part of it. And then you have deep learning, which I'm not going to cover, but machine learning is it. So in an artificial intelligence and machine learning, we can ask the machine to find out which is the uh, which image is the correct cat. Like in this case, we have a cat, so we can ask whether is it a cat or is it someone else, some other animal, say for example a dog or a rabbit or something like that. So basically, we are just giving them inputation and asking for their validation, whether it is this or it is that. Then come to the next topic. Now, artificial intelligence, what are the advancements in machine learning and AI in medical microbiology? So basically, let's go to over here. So we can ask the machine learning in medical data that it can predict what is the likelihood of the patient in developing a new disease. So in developing a certain disease also can be helpful in it. So making sure we can just help them improvise the diagnostics and find out what test is very particular to it, so that we don't have to do unnecessary tests and wasting a lot of money and all the resources of the patient. Thus helping out, being very particular and being in a singular time itself. And the third topic is machine learning in AI and in pathogen detection. So we can using over here, we can ask the machine learn to analyze genetic sequences in the microbes, wherein each microbe has its own genetical sequences. So we can ask the AI to look into the genetic sequences and help us find out new pathogens, since we have antimicrobial resistant bacteria which are being produced right now, and we have viruses which are completely advancedly mutating on its own. We can ask them what is the pathogen that is causing this patient such pain and causing infection, and we can ask them or learn from the machine itself how does it behavior, like how does it change on its own, what are the things that is required them for the changes, and how we can stop it off or just cut it off itself. So making us understand, okay, this is pathogen, and how can we just undo it from replicating on its own in a behavior. Now coming to the fourth one, that is machine learning in disease diagnosis. So we can ask a machine learning as a machine to analyze the X-rays and MRIs to identify the early stages of cancer and other diseases. Since machine learning can, if we give them enough input, like this is the core information that we have right now, and can you replicate it and find out what is the, whether it is a cancer, if it's a benign, or is it a malignant, or anything, or is it a cancer and on its own case. So we can ask them. So it can help us understand whether we are in early stages, or are we in the late stages on its own? So we can, using AI, we can really fasten up the speed, and we can early on detect whether any patient has a say cancer or any other disease, which we can have, reverse it back to the patient, reverse the infection back, causing no more pain, no more further spreading of the infection on its own. And now coming to the machine learning and AI and drug discovery. So with using machine learning, we can ask the AI to find out what and predict the efficiency of a particular drug, uh, and also suggest any other drug which can be helpful in helping in this infection course of due of time. Well, and also repurpose existing, that is, uh, say for example, we have an XYZ drug, and it has to be targeted on a particular portion. Say for example, the core, whether the bacteria or a organism is replicating. How about we just stop it from the starting itself? Best of all. So we can ask the AI, like, can you just suggest something, or can you just repurpose the drug by changing it or to some of its content, which can help us in in cutting off the infection on its own and at the starting itself. That is the best part. Next, yeah, coming to the sixth point, that is in personalizing medicine. So say for example, if a patient is say for example, a patient has a cancer, of a lung cancer or something, and he has to go to a chemotherapy. So we can ask the machine learning to analyze the genetic data of the patient. We all are having DNAs we are not common DNAs on our own. So we can ask the machine learning to look into our genetic system and find out what is the best treatment for our own, and suggest finding out the most efficient option which we can help in personalizing a particular medicine for our own benefit, and making it not like causing any side effects, because each drug has its own side effect on its own. So if you personalize the drug on our own genetic data, we can limit the side effects which are being caused, helping us without causing any unnecessary let's just say pain and all that thing itself. Now coming to the seventh point, that is machine learning in detecting pathogens. So we can ask the machine learning to go through all the, if we place we can ask the machine learning to analyze the data. Say for example, we recently had a pandemic, that is COVID-19. So we can ask the AI to analyze data from the sensors we can place in the public areas. We can help, which can help in finding out the presence of the airborne pathogens which are being present. So if let's just say uh, if let's just say in an XYZ place, we place a sensor, and in that place around which has a crowd, and we can sense that there is around 54 to 65% of the COVID-19 pathogens which are present in that area. We can close that area, and we can place a lockdown or something like that, preventing people from moving around that area, and advising people to carry a proper precautions, making sure it doesn't spread on its own. So we can use the AI machine to find out what is the efficiency, where can we find out that this is such infections and all other, where airborne pathogens are present in that area, in an XYZ area. Coming to the eighth one, that is in diagnostic, we can ask same as as I said earlier, since since the celebrities or since any any people, we have very life-threatening infections such as Alzheimer's infections or a cancer or infectious infection that causes life-threatening infections, which can lead to inevitably death itself. So we can ask the machine learning how can we can understand what are the diagnostics, like how can we understand the pattern, like what it causes, so that we can prevent this infection from having to another person itself. say for example, if we have a patient who is about his son is that bad, so we can take a sample of it and ask the machine learning identify the course of it, identify how this infection has occurred, so that we can prevent it from happening to any other person on its early stages itself. Thus, helping us, prevent, making sure that most of the patients, or as much as we can, doesn't have this infection on its own. So limiting this infection on the course due of time itself. And also in discovering new drugs, as I've said in my third or fourth point, it said that you can just make sure that we can discover new drugs, irrelevant to the old drugs itself, which can cause side effect, but we can discover new drugs which limits the side effects, thus helping in the efficiency of the drug, and also in making sure that the microbes, the antibacterial bacteria doesn't cause any resistance to the new drug, since we have to behind a drug which is sensitive towards a particular bacteria or a particular bacteria itself. So thus, we can discover new drugs and making sure that yes, so we can make sure that it is not resistant to a certain intake, to a certain intake. And lastly, we can ask the machine learning saying, study a particular risk of it, like find out what is the risk of a particular drug, and helping out in personalizing medicines, as I said before. Basically, asking them to reduce the risk of this infection being, further spreading out. That's what my point is. Now, coming to the second part of it, that is, what are the challenges and the limitations? So we have a couple of challenges. First one is limited computerization resources. Since we are, say for example, we are in a hospital, we don't have that much of resources, say for example, that much place or that much amount of electricity that is required for this machine learning to work on its own, because they are supercomputers. They require a lot of energy, they require a lot of data to be analyzed to make sure they come to a proper conclusion. And coming to this conclusion, we have limited resources, since in a healthcare, we can't afford that much of, say for example, if high-end hospitals and all are there, they can acquire it, but small hospitals and all small clinics, that's not possible. But to some extent we can, but it requires a lot of resources, and we can't fulfill all the resources on its own and on all to a certain intake. So yes, these are one of the limitations. And coming to the next one. Yeah, coming to data sources, acquisition and storage. A computer requires a lot of data, say 1 terabyte, 2 terabytes, and machine learning and artificial intelligence, it requires a lot, 4 terabytes to 3, as what I've understand and I heard. So acquiring this so much of data and storing it so that it doesn't, what to say, get encrypted, or it doesn't get duplicated, or get leaked out in any way. So storing that much of data is very critical, and it requires a lot of machineries, as what I said. Making sure each and every hardware is there in proper thing, and there's a lot of manpower also required. Because in any way, if say for example if a hard disk gets shorted up, or it's not working, so all the data that's present on it is lost. We can prevent it by keeping in cloud like Google Drive or like high-end software like that, but it requires a lot of expenses on its own. So yeah, this is also one point which is causing a lot of limitation and challenges. Then coming to the lack of
sorry to interrupt you. The time has been provided for you. It's 10 minutes. Please try to conclude it as early as possible.
Yes, coming to lack of high quality and diverse, because machine learning requires a lot of data on its own, and with the lack of data, we can't come to a proper conclusion also. Let's come to integration. Since machine learning derives data from different sources, it can be from a known source also. So making sure we don't take it from any XYZ illegal sources, it can be a ethical problem also on its own. Coming to data privacy is important since we have a lot of data being leaked online itself on the net, and confidentiality is important because in a healthcare we try to keep all the information in confidential so that it doesn't leak out. Preventing this is one of the cases because hackers and all they try to buy bleach the data, and they try to take the data and try a ransom for it. There are clinical limitations to it. It's not like there's infrastructure. As I said earlier in the point, resources, manpower is required. A lot of equipment is required on its own. And yes, a lot of technical people are required for it. Coming to resistance, yeah, there are a couple of doctors which are resistant to it. They are like, say for example, an experienced doctor says like, we don't trust this, software, we don't trust this AI. So yeah, there's a lot of backlashes on it. And coming to limitations in medical microbiology, it's not like it can offer a potential benefits to some extent, but yeah, there are limitations on its own. So, for example, in antimicrobial this thing itself, there are limitations. I think. Let's come to ethical. That is that, for example, in potential patients and all, we try to keep things in a proper manner. But there are like something called the data bias and all that in an area. Now, coming to the that is a current applications for it. So right now we are using it for quick development and quick identification and the presence of samples in pathogens in clinical samples. Right now they are being used. Yes, that's one thing. And one thing for sure that is we are developing new antibacterial agents, that is antibacterial drugs, which can be helpful in making sure the resistance is very least on its own to a particular infection. Next comes the third point, that is development of new vaccines. Yes, vaccine. We did use AI for the process of a new vaccine, and yes, make sure the immune response is very high to a particular infection. That's the thing. Coming to the improvised disease prediction, we can predict, as I said before, using AI. The current application is that we feeding its data and making sure it can predict us the likelihood of the infection and what is the next stages which it can go to the next thing, and better understanding the microbial pathogens, finding out what is the mechanism, how it replicates on its own, how does it transfers, and what is the way that we can make sure it doesn't travel to its own. Coming to the second last the future developments and vision. As I said before, it's making sure we integrate it and analyze the data and helping medical professionals like me and doctors and nurses in making sure advancing in imaging so that we can identify what is the diagnostic imaging saying to us, and making sure that we can find out what can be the better option for it. Also helping us integration in healthcare and AI. So we can identify the particular patterns and make sure that if, let's for example, in the next future years from here, we can prevent further pandemic which can occur. As what I've heard, a fungal pandemic can occur in future years. So we can prevent it so that, and we can also find out if it's happening, what are the chances we can do to, further transfer it also, transfer. Then coming to the last thing, in microbial research also, we can use it in research field itself so that we can find out what is the best options for it. Now come to the last part, that is conclusions. So in the conclusion summary, I would say that AI on its own is one of the best. Is in its clinical starting stages, but it is helpful. It is helpful in finding out the basic identification of the bacteria or a virus or a fungal which occurs in a patient, and finding out what is the treatment best for it, and how can we help it in making sure it doesn't transfer and it doesn't replicate on its own. Second point is discussion. We have to discuss with a lot of software ethical person, along with a lot of people to find out whether it is very helpful really in our scenarios, and is it within our budget also. And calling for action, we have to make sure that most of the people which are on board to this particular AI in a healthcare.
Selvin, sorry to interrupt you, but you're out of time already. Session chairs, any questions for him?
Selvin?
Yes, ma'am.
Very good presentation.
Thank you, ma'am.
As I don't know much about the artificial intelligence and machine learning, but I want to learn more about this because it is a very interesting topic. You have discussed about the pathogens and about the disease and even about the drug discoveries. Can we use the artificial intelligence and machine learning to predict the antimicrobial resistance, which is a major concern?
Yes, ma'am, absolutely. I think so. You have told in the application part or somewhere on that part, we can place some sensors. Yes, we can find out how this can transfer also, so that we can find out what is the main transmission mode, which the microbes are using for their advantage, and make sure we cut it off on its own, so it doesn't transfer, and preventing the transmission and transfer of this infection to a large area itself, which can cause an epidemic on its own in any XYZ area.
Okay. One more concern. Can you share your PPT?
Yes, ma'am. Absolutely. I will share my PPT.
So please write down my email ID and please mail me your PPT.
Yes, ma'am. Just a minute. Yes, ma'am.
It's Muktavats, MUKTA. MUKTAVATS @yahoo.com.
Is it correct, ma'am?
Hello, am I audible?
Yes, ma'am. You're audible. I'm sending to you right now itself.
MUKTAVATS @yahoo.com. Okay.
Yes, ma'am.
Okay. Thank you so much.
Yes, ma'am. You're welcome.
Srijit sir, any questions from your end?
No, thanks. You're already over your time, so I don't want to ask any question. Thank you.
Thank you. Thank you, Selvin. The other presentation.
Thank you very much, ma'am.
Yeah. The other presentation is by Mustafa from Lovely Professional University, going to give his oral presentation on screening of Pseudomonas aeruginosa clinical isolates for antimicrobial resistance and biofilm production.
Am I audible?
Yes, you are audible. But before going to the presentation, here is the instructions for the participants. The time that is provided for each participant for oral presentation is 10 minutes. 8 minutes is for presentation and 2 minutes is for discussion, question and answer session. So I request all the participants to stick to their time and complete the presentation before that. Thank you.
Is my screen visible?
Yes, it is visible.
Hello, respected panels, moderator, and my dear colleagues. Myself, Mustafa Vora. I'm a PhD scholar from Department of Medical Laboratory Science, Lovely Professional University. And I'm going to present on the topic evaluation of Pseudomonas aeruginosa clinical isolates for antimicrobial resistance and biofilm production. Okay, 1 minute, please. Yeah. So, what is biofilm and biofilm associated infections? So, biofilm is a assemblage of surface associated microbial cells that is enclosed in an extracellular polymeric substance. You can call it as a population of microorganisms working as a single unit. Why do microorganisms form biofilms? Because of some external triggers such as lack of nutrition or exposure to certain inhibitory substance. The microorganism transforms from planktonic form, which are individual moving, to a group of population of microorganisms, which is termed as biofilm. Now, why do they do this? Because the morphogenic switching of microbes from planktonic to biofilm gives them certain phenotypic advantages, such as tolerance to various chemicals, desiccations, efficient use of limited substrate. So thus, whenever the conditions are favorable, microorganisms usually do try to survive as a biofilm. Now, for us, it is very harmful because it is associated with various infections such as ventilator associated pneumonia. Moreover, the biofilm associated infections are chronic in nature. That means they are recurring and they persist for a long interval of time. Also, the virulence and pathogenicity of most of the clinical isolated are closely related to biofilm formation, and hence it is necessary to understand the process of biofilm formation. So here I am showing one of the examples that biofilm is closely related to antimicrobial tolerance. When a biofilm is formed, there are various ways the microorganisms are not affected by antimicrobial. Various mechanisms. One of the major mechanism is the poor penetration of antimicrobial. Because of the polysaccharides form, the antimicrobial cannot penetrate the biofilm and hence attack the microorganism. Also, in biofilm, there are different types of microorganisms based on their metabolic activity. So many of the microorganisms in biofilms are persisters, which are metabolically inactive, and hence many of the antimicrobial cannot affect biofilm which are antibiotic, which are associated with growth of cells, and hence such persisters microorganisms are not affected by such antibiotics. Now, what was my aim of this study? The aim was to study the biofilm production of Pseudomonas clinical isolates. Most of the study of biofilm formation is done using standard strain PA01 and PA40. Pseudomonas is a harmful nosocomial clinical isolate, which is known for its biofilm formation, and hence it was important to understand biofilm production in clinical isolate. Another aim was to understand the antimicrobial resistance in the Pseudomonas clinical isolates, and whether and find whether there is any correlation between the antimicrobial resistance and the biofilm production among the isolates. So methodology, simple methodology was used. Pseudomonas aeruginosa was isolated from clinical samples, and it was further purified and used, and antimicrobial resistance was determined using Vitek or distribution method. This was the workflow. The clinical isolates were isolated Pseudomonas aeruginosa from clinical samples. Then antimicrobial resistance pattern was determined. Then its biofilm formation was checked using different phenotypic method and on different surfaces. And finally, the biofilm related gene expression was checked using RT-PCR. Now, this is the antimicrobial resistance pattern of the overall clinical isolates. Around 80 isolates were used for this study. And most of the, as seen from the antibiogram, that the most of the organisms were quite sensitive to colistin, gentamicin, amikacin, and meropenem, and they were resistant to ceftazidime, ciprofloxacin, piperacillin, and tazobactam. Now, crystal violet was used for determining biofilm formation. In crystal violet assay, microorganisms are grown in 96 well microtiter plate and incubated for 24 hours. After incubation, the planktonic cells are removed and then it is stained with crystal violet. After staining with crystal violet, it is destained and the absorbance is read in a reader, and then the amount of crystal violet relates to the amount of biofilm formed on the microtiter plate. So this is the photo of the biofilm grown on the microtiter plate. These are the results of the crystal violet assay. As you can see, many of the microorganisms, the biofilm formation was also checked along with the control. So as compared to control, there are many biofilm, high biofilm forming organisms, and majority of the microorganisms were forming biofilm. This is a graph that we can see that we can divide the biofilm formation into 3 different categories: strong, moderate, weak, and none, which do not form biofilm. So most of the clinical isolates were found in moderate category. There were some pathogens which were forming high biofilm. This is a graph showing the different type of biofilm forming producers among different clinical samples. As you can see, pus, swab, and urine were the majority of samples where pathogens were isolated and different category of biofilm formation was seen. Interesting is sputum, where almost all the biofilm formation were of moderate category. As we know that Pseudomonas aeruginosa is known for a cystic fibrosis, so this could be a very good indicator of understanding biofilm formation from sputum sample and knowing the cause of the infection caused by the Pseudomonas aeruginosa. Biofilm formation was also grown on different types of surfaces such as latex, catheter, slides, glass slides, polystyrene Petri plate. This was on a polystyrene tube, and this was on a PEG lid. Now, this is an interesting methodology in which we can not only observe the biofilm, but we can expose it to different antimicrobial. This is the method for growing biofilm on a PEG lid, where we grow the biofilm in a liquid and we put a lid and allow the biofilm formation to grow on a lid. We can also challenge it with antimicrobials and check what are the which organism or which isolates is showing inhibition to certain type of drug. So using all these surfaces, the reason for this study was that usually study is done on only one surfaces, but as read on the as it is found in literature, the biofilm formation is dependent on which surface you grow. So if you grow on a polystyrene plate, you may get a strong biofilm, but if you grow on a catheter, you may get a weak biofilm formation. So selected strain were studied for biofilm formation of different surfaces, and categorization was done, and few strains were found consistent to form, such as PS30, where PS30 or PS104 was found to form consistent biofilm in different types of surfaces, while organisms such as PS51 and PS45 were found to be consistently weak in forming biofilm. So these are we can study such type of organism as why they are consistently forming high or low biofilm on different type of surfaces. Now, in order to understand the biofilm formation, its gene expression is also needed to be checked. That what are the so for that 22 strains were selected and RNA was extracted, and expression of various biofilm related genes such as alginate, lactinase, PAL polysaccharide, and PSL polysaccharide was checked. 16S gene was used as a reference gene, and delta CT was calculated. So here you can see the graph of differences in delta CT value of different biofilm related gene of selected isolates. So most of the organism except the 2 organisms were found to form alginate. Now alginate is responsible for mucot type of colony. So we can understand that this organism must not be forming a mucor type of colony. Similarly, LAC-A, there was variation in different type of LAC-A expression, and PALE. PSLA was not detected, as we later found that there is error in the primer formation, and hence we could not detect PSL in any of the isolates. So various biofilm expression was checked. The reason to check biofilm gene expression is that whether the phenotypic method and genotypic method correlates. If we get a high biofilm in phenotypic method, do they also form high biofilm? Do they also have a high biofilm gene expression? So in summary and conclusion, we found that majority of isolates were a biofilm producer and also demonstrated high expression of various biofilm related genes. 30 isolates were further studied, and it was found that overall strength of the biofilm was similar on different type of surfaces. Slight differences were found, but most of them were similar. But in addition, it was also found that there was no correlation between high biofilm production and antimicrobial resistance. There is a quite contrasting literature where it is also found that high biofilm production has lead to antimicrobial resistance, but there are many literatures which have opposed this correlation. In our study, we didn't found any correlation between high biofilm production and antimicrobial resistance. I would like to acknowledge my guide, Dr. Sandeep Sharma, LPU Punjab, and my co-guide, Dr. Narayan Kamath from BBCH. Thank you.
Thank you, Mustafa. Session chairs, any questions for him?
Hi, Mustafa.
Mustafa, very
Good presentation.
Thank you, sir.
So I was wondering, so when you measured the biofilm related gene by qPCR,
yes, sir.
what was your internal control?
My internal control was 16 as a gene. I was using as a internal control.
16 is rRNA.
Yes, sir.
Okay. And like a more general question, so like why some bacteria are good at biofilm formation than others? Do you have any comment?
That is the question. That is the thing which we still don't know, that what is the mechanism. Why certain organisms do fire biofilm? What are there any genetic components responsible actually for biofilm formation, or whether just an environmental condition can trigger the common genes which are found? So my overall major study is that this was the initial objective. That only I'm trying to study that why certain organisms form low biofilm, why some organisms are forming high, and why some are showing moderate biofilm. That is still yet to be found. Concrete proof is yet to be found that why it is some are biofilm and some are not.
Okay, thank you.
Thank you.
Mustafa?
Yes, ma'am.
I have a very simple question. You have talked about the genotypic characterization and phenotypic characterization. I just want to know, how will you identify the Pseudomonas by using a traditional microbiology? It's a very simple question.
Biofilm formation.
No. Identification of the Pseudomonas by using traditional microbiology. By using the traditional microbiology.
By culture, we get the pigmentation, and by oxidase test we can, and by gram staining we can specify that okay, this might be Pseudomonas aeruginosa strain.
Can you explain the oxidase test?
Yes, Pseudomonas, the oxidase organisms use certain enzymes, oxidase enzyme, which they can tolerate certain anaerobic condition. So Pseudomonas organism is an anaerobic, not obligate anaerobic. It's an facultative anaerobic organism. So this organism has that oxidase enzyme, which it can tolerate both oxygen level, high oxygen level, and hence when we take the swab and put it on an oxidase strip, its color changes because of the presence of enzyme. The color changes to purple.
Which dye we use in this test?
I'm sorry, ma'am. I don't
Name.
remember.
It's TMPD, tetramethyl paraphenylene diamine dihydrochloride.
Okay, madam. Sorry,
Okay,
I don't
thank
remember.
you so much. Very good.
Okay,
Very good
thank you.
presentation. Thank you.
Thank you, ma'am. Yeah.
Thank you, session chairs. We are going with the other presentation of oral by Karpagavali from National Institute of Siddha on topic a review on antimicrobial activity of herbs used in Siddha system of medicine. Karpagavali, are you available with your presentation? Karpagavali, can you hear me? Okay, we are moving over to another presentation by Ankita from Senkarlal Kandelal Arts, Science and Commerce College on the topic of screening of Ayurvedic drugs against multi-drug resistant Staphylococcus aureus. Ankita, I hope you are available for your presentation.
Hello, ma'am.
Yeah, Ankita, we can hear you.
Okay, ma'am. Can I start?
Yes, you can start with your presentation.
Hello, everyone. Good afternoon to all. My name is Ankita Lokhandi, research scholar from Department of Microbiology, SPC College, Akola, Maharashtra. Today, my presentation topic is screening of Ayurvedic drugs against multi-drug resistant Staphylococcus aureus. Antimicrobial resistance is nowadays a major trouble to patient treatment. It results in expanding morbidity and mortality, prolonged hospital stays, and significant economic loss to the patient and nation. Diseases and infections caused by antibiotic-resistant strains of S. aureus have reached epidemic proportions around the globe. There is emergence to replace therapies for the treatment of drug-resistant infections and to focus on least use of antibiotics to reduce the development of antibiotic resistance. Ayurveda is
we cannot start screen sharing while the other
Hello.
Participants, I request everyone to mute their mics. A small request to all the participants. I request everyone to keep their mutes mic so that the session remains undisturbed. I hope you'll understand this and cooperate with me in remaining silent. Yes, Ankita, you can go with your presentation now.
Okay, thank you. Ayurveda is a traditional medicine of Indian history in contrast to allopathic medicine, which primarily uses synthetic chemicals designed for specific target receptors and provides symptomatic relief. Present research work is aimed to search effective Ayurvedic drugs and their combination against multi-drug resistant Staphylococcus aureus. As compared to modern science, there is a list research is done on Ayurvedic drugs as compared to allopathic medicines. So there is need to focus on Ayurvedic drugs and develop new strategy to combat multi-drug resistance by using Ayurvedic drugs to increase their sensitivity and to treat infectious diseases with the help of effective formulations. The hypothesis is antibacterial Ayurvedic drugs may inhibit multi-drug resistance Staphylococcus aureus. The aim of this study is effect of Ayurvedic drug against multi-drug resistant strains of Staphylococcus aureus by screening of Ayurvedic drugs. And the objectives are to isolate S. aureus from suspected clinical samples, to identify bacterial isolates and study of their antibiogram, to check the sensitivity of drugs, and to study antibacterial activity of some Ayurvedic drugs against the isolates of multi-drug resistant S. aureus. Now, materials and methods. Clinical samples of patients suffering from S. aureus infections was collected from labs in Akola, and enrichment carried out by using trypto-soya broth. A loopful from enrichment medium is trichone nutrient medium like mannitol, salt, agar, and incubated at 37 degrees centigrade. Isolates was studied for microscopic, morphological, cultural, and biochemical characteristics. Isolates was studied for their susceptibility pattern to commonly used antibiotics by performing antibiotic sensitivity test as per Kirby-Bohr method using Muller-Hinton medium. For screening, Ayurvedic drugs having applications of infectious diseases was purchased from market. Drugs and their extracts like methanol, ethanol, acetone, and aqueous extract was tested for antibacterial activity against clinical isolates of S. aureus by performing antibiotic sensitivity test as per Kirby-Bohr method using Muller-Hinton medium. Now, result and discussion. In present study, antibiogram study showed that gentamicin, rifampin, vancomycin, ampicillin, and cefazolin have inhibited the growth of strain 1 and 3 of S. aureus, while for strain 2, S. aureus only strain 2 of S. aureus only vancomycin showed antibacterial activity. Table number 1 shows the zone of inhibition, that is antibiogram study. In the present study, the samples namely Kadu Neem, Manjishta Churna, Nimpati Churna, Bihada Churna, Arjunsal Churna, Trikatu Churna, Dhatake Churna, Jati Churna, Marit Churna were tested for their antibacterial properties against the 3 strains of Staphylococcus aureus. Result shows that acetone and ethanol extract of Kadu Neemachal Churna possesses antibacterial activity against 3 strains of S. aureus, while methanol and aqueous extract showed no activity. Kadu Neemachal Churna has various clinical applications, antibacterial, antiviral, antidiabetic, and anti-cancer properties. Ethanol extract of Manjishta Churna possesses antibacterial activity more against strain 2 of S. aureus as compared to other strains, while ethanol, acetone, and aqueous extract showed no activity. Manjista is used as an anti-acne. In ancient time in India, it is used as a blood purifier, hence it mostly used against blood, skin, and urinary diseases. Ethanol extract of Neempati Churna possesses antibacterial activity, while methanol, acetone, and aqueous extract showed no activity. Methanol, acetone, and ethanol extract of Bihada Churna showed antibacterial activity, as well as aqueous extract shows antibacterial activity only for strain 2 and 3 of S. aureus. Aqueous extract of Arjunsal have no activity, otherwise ethanol, methanol, and acetone extract shows antibacterial activity. Arjunsal have mainly used to cure earache or as a laxative, anti-dysentery, anti-cancer, antiviral, etc. It shows a good activity against ear pathogen. This table indicates the zone of inhibition of Ayurvedic extracts against the 3 strains of S. aureus. Only methanol extract of Trikatu Churna possesses antibacterial activity. Dhataki Churna showed antibacterial activity in methanol, acetone, and ethanol extract, while no activity in aqueous extract. The flowers of Dhataki contain self-generated alcohol, which helps to reduce wound contraction. Methanol, ethanol, and acetone extract of Jati Churna showed antibacterial activity, while aqueous extract showed no activity. Ethanol extract of Marich Churna showed no activity, while aqueous, methanol, and acetone possesses antibacterial activity. Marich, that is a black pepper, showed good antibacterial activity. Papain, terpenes, and flavones used to inhibit the growth of antibacterial pathogens. Next is summary and conclusion. Emergence of resistance against commonly used antibiotics has become a serious global problem. There is need to develop new strategy of using Ayurvedic drugs against infectious diseases. Extracts of Ayurvedic drugs and medicinal plants have been tested against multi-drug resistant S. aureus, but intensive research is required to know the application potential of Ayurvedic drugs and phytochemicals and essential oils and their combination for treatment of infection caused by multi-drug resistance S. aureus. Our findings suggested that Ayurvedic preparations extract have potential as an antibacterial activity against pathogenic bacteria S. aureus. They can be used to treat in a treatment of infectious diseases. The data obtained in this study indicates Ayurvedic preparations are good to inhibit the growth of strains of S. aureus. It concludes that Ayurvedic preparations is used to prevent and control infectious diseases. These are references. Thank you.
Yeah, thank you. Thank you for your presentation. Session chairs, now over to you for the question and answers. Mukta ma'am?
Yes, ma'am.
Can we have any questions for Ankita? Dr. Mukta ma'am? I hope ma'am is having some technical issues. Srijit sir. Okay, thank you, Ankita. Thank you for your presentation. We'll be going for the other presentation. Now, the presentation is by Sandipta Debanath from Sister Nivedita University. Is going to give oral presentation on nosocomial infection.
Yes, ma'am.
Yes, you're audible and your screen is also visible. You can go with the presentation.
Hello. Hello, ma'am.
Yes, Sandipta. Sandipta, we can hear you.
Okay, ma'am. Ma'am, can you see my screen?
Yes, yes, we can see that.
Okay, thank you. So, good afternoon, everyone, and good afternoon, every teachers and professor and all faculties present over here. I am Sandipta Debanath from Sister Nivedita University, and my co-author is such a Atya Ghosh, our professor from microbiology department. So today I start to discuss the nosocomial infection. Sorry, ma'am.
ഞാൻ നെറ്റ്വർക്ക് റിസോഴ്സ് സെന്ററിൽ കയറി ഇരുന്നോട്ടെ എനിക്ക് പ്രസന്റേഷൻ ഉണ്ട് താങ്ക് യു
Sandipta, are you there?
Yes, ma'am. Yes, ma'am. My seriously is problem.
We cannot see your screen.
Now you can.
Yes,
Ma'am,
yes. Now we can see.
So what is nosocomial infection? Nosocomial infection, which is also referred to as healthcare associated infection, or the infection acquired during the process of receiving healthcare that was not present during the time of illness. Hundreds of millions of patients around the world are affected by the healthcare associated infection every day. HIV result in prolonged hospital stays, long-term availability, and increased resistance of microorganisms to antimicrobial, massive additional cost for health system, emotional, personal burden for patient and their family. Finally, unnecessary death. To prevent HAI or nosocomial infection, it is important to identify their cause, understand how big the problem is, and evaluate whether the intervention is reduced HAI are successful. So now we are discussing this all. Nosocomial, the word comes from the nosos, disease, and comion, the to take care. It leads to reduce the quality of time and may even lead to death. They may occur in different side of healthcare system, as in long care facilities, ambulatory setting, and may also appear after discharge. The intensive care unit, ICU. The ICU is the barn of nosocomial infection. Due to its unique environment, use of various antimicrobial and invasive monitoring device. So here we can see the nosocomial infection in a single page. First of all, you can see presence of microorganisms in hospital environment. This is the most root cause of nosocomial infection. For this, the patient are immunocompromised, and transmission of this pathogen between staff and patient among the patient. Now, types of healthcare associated infection or nosocomial infection. The Central Disease Control and Prevention Board categories the type of HAI as follows. 1. Central line associated bloodstream infection, catheter associated urinary tract infection, surgical site infection, ventilator associated pneumonia, non-ventilator associated hospital acquired pneumonia, gastrointestinal infection, including Clostridium difficile and urinary tract infection. Also, ear, eye, nose, throat infection, lower respiratory tract infection, cardiovascular infection, central nervous system infection, and reproductive infection, and many more. Here we can see a chart. The data which too backdated, but now it is rise gradually. So surgical site infection, we can see that 33.7%, it is such a low value. And ventilator associated infection, 31.6%, central line associated bloodstream infection, 18.9%, and C difficile infection, 15.4%, and catheter associated urinary tract infection, 1%. Now the causative organism. The three main causative organisms are bacteria, fungi, and viruses. Bacteria. Now come to bacteria. Bacteria are gram positive and gram negative. Gram positive Staphylococcus aureus, coagulus negative Staphylococcus, Streptococcus species, Enterococcus species, just like Phaeacilis phaeaceum. And gram negative is Klebsiella pneumonia, Klebsiella oxytosa, Extida coli, Proteus mirabilis, Pseudomonas aeruginosa, and Acinetobacter pyomen. Fungi, Candida albicans, Candida parapsilosis, Candida glaberta, and Candida aureus, and Aspergillus fumigatus. Viruses are hepatitis B, C, and human deficiency virus. The most dangerous rhinovirus, rotavirus, cytomegalovirus, herpes simplex virus, and influenza. Here we can see what is mostly infected in the nosocomial infection. Escherichia coli, E. coli, the most infected in nosocomial infection. Then Staphylococcus aureus, Enterococcus species, which is 10%. Then Pseudomonas aeruginosa, Klebsiella species, negative coagulus Staphylococci, Candida species, Clostridium difficile, Enterobacter, and Proteus species. This is a small chart to describe what are the ratio of this bacteria to cause nosocomial infection. Now, this is an epidemiology of nosocomial infection in male patient and female patient in the worldwide. Male patient, we can see that male patient is mainly infected in SBP. Then, where the female patient is mostly infected by UTI and SBP both also. And pneumonia, this male patient is mostly infected, but female patient is low. Bloodstream infection also and the same like an infection without identifiable source, that is mostly infected by female, and other infection both are almost same, but a female are 14% and male patient are 9%. Now this is a chart in the rest of Asia specific, China and India, how nosocomial infection now the data, the small data. We can see here the increase, how is gradually increased day by day. 2012 to 2022. Nowadays, India is mostly dangerous position. Now, this is the hospital scenario of India. That globally the accepted rate of HIV is up to 8%, but India it is already 18%. In presence of Premier Institute of AIMS and SGPGI, it's around 20%. In government hospital of UP, it's the worst condition that there are 40% are HIV patients. And for the and if we see in 100 patients, if we break down in the nosocomial infection, we can see the 40% urinary tract infection, 25% surgical site infection, and 7 to 10% which infections such as septicemia and ventricular associated pneumonia, 1.5 to 5%. Now complication. Complications of HAI are broad and depend on the type of infection, the severity of illness, and implicated pathogen. First, hospital acquired pneumonia (HAP) and ventilator associated pneumonia (VAP). The complication are respiratory failure, emphysema, paraneumonic effusion, sepsis, ABSI, which have a central line associated bloodstream infection. Then its complication is spurative thrombophlebitis, endocarditis, septic arthritis, abscess, and sepsis. Then CAUTI. CAUTI is mixed as a catheter-associated urinary tract infection, upper urinary tract involvement, and sepsis. SSI, surgical site infection, which is a delayed wound healing, rejection of implanted device, prosthesis, repeat surgery or removal of infected device, abscess formation, body cavity infection, and sepsis. And then hospital acquired C. difficile infection, HOCDI, recurrent or difficult to treat infection. It is the most complicated and ileus with toxic megacolon, dehydration, and sepsis. Then come to pathogen transmitters. How it transmit? It is transmitted by various means: indirect, direct, just like medical equipment, implant, and device. All that is present over hospital and the contaminated medical staff and visitor and hospital facilities, which is very ruined nowadays in government hospital. Then diagnosis and treatment. How we can diagnose it? Many forms of nosocomial infection can be diagnosed through the site alone. So only we can see it from gradually, and we can diagnose it. That's which type of nosocomial infection is this. Then pools, inflammation, and rashes may be indication of infection. Blood and urine culture test can be identify the infection. Treatment for nosocomial infection depend on the infection type. What is this infection? Is this SSI? Is this DAP? That's depend on it. We can treat it. If the infection
Mr. Sangita, are you facing any technical difficulties? I hope Mr. Sandipta is having some technical issues. We are moving with the other presentation. Oral presentation by Mohan Kumar from Chettinad Academy of Research and Education on the topic, a study on the bacterial flora, including pathogens on the hands of fishmongers.
Hello, good afternoon, ma'am. Am I audible?
Yes, you are audible, Mr. Mohan Kumar.
Ma'am, can I share my PPT?
Yes, you can.
Thank you.
Hello, ma'am.
Sandipta, can we have your presentation after Mohan Kumar? You can resume.
Ma'am, my microphone was lost. Okay, ma'am.
Hello, ma'am. Is my screen visible?
We cannot. Yes, now we can see Mohan Kumar.
Can you start my presentation?
Sorry, your voice is breaking. Yeah.
Can I start the presentation, ma'am?
Yes, you can start. But before going that, Sandipta, after completing Mohan Kumar presentation, I will be calling up you. Please make sure that you're having with good connectivity, okay? Yes, Mohan Kumar, you can go with your presentation now.
Thank you, ma'am. Good afternoon, everyone. Myself, Mohan Kumar. I'm a research scholar in microbiology department, and our co-speakers are Mr. Harshit, BSc Allied Health Physical Laboratory Technology, and our Dr. Priyadashini Shanmugam, ma'am, Professor and Head of Microbiology, Chettinad Hospital and Research Academy of Research and Education, Tamil Nadu. In this session, we would like to do a presentation on bacterial flora, including pathogens on the hands of fishmongers. Table of contents. These are the contents that we're going to see now. Introduction. Bacteria are tiny single-celled organisms that are found almost everywhere, including the human body. Hand is an important source of infection as when compared to other parts of the body. Contaminated hand plays a major role in fecal oral transmission disease, and hands of fishmongers can carry different types of pathogenic organisms compared to other workers. And this can lead to food poisoning, diarrhea, and other bacterial infections, both for themselves and others. Aim of the study: to isolate and identify the microorganisms from the hands of fishmongers and control group. Objective of the study: to collect hand swabs from fishmongers willing to participate, to isolate and identify the microorganisms from the hand swabs by performing various culture methods and biological reactions, and to determine the pathogenic bacterial prevalence in the hands of fishmongers and control group. Methodology. This is a prospective study, which is Chettinad Academy of Research and Education. The study period February 2023 to end of March 2023. And for this study, we have taken 50 hand swab samples from the fishmongers and 20 hand swab samples from the control group. And for this study, the control group peoples were housewife, homemade students, and elder people. And inclusion criteria. Individuals who work as fishmongers were included in the study, and individuals should not be suffering with any skin disease, inflammation, or irritation. And our exclusion criteria are individuals with other occupation and all the individuals with any skin disease, inflammation, or irritation. Methods. This is the method followed here for processing the collected samples. As per the samples, collected samples were inoculated on the nutrient agar. Then the nutrient agar were putting it to the incubator for overnight incubation. And once the colonies were grown on the culture medium, first we did the preliminary test. And simultaneously, we subcultured the colony from the nutrient agar on enriched and differential media. And we did the biochemical reactions along with that. And the results were interpreted on the next day. As I said, first, we have fishmongers and control group. All of the hand swabs were collected from the fishmongers of Kovalam region. And this sterile cotton swab container were used for collecting the samples. Once the samples were collected, we directly inoculated on the nutrient agar. And this is how the colonies were looking on the nutrient agar after overnight incubation at 37 degrees Celsius. Out of our 50 fishmonger hand swab samples, 6 of the samples were showing 3 different types of colonies on the nutrient agar after overnight incubation, and 24 samples were showing 2 different types of colonies on the nutrient agar, and the rest of the 20 samples were showing only 1 type of colonies on the nutrient agar after overnight incubation. On the other side, in the control group, out of 20 samples, none of the samples were showing any 3 types of colonies on the nutrient agar, and 13 samples were showing 2 different types of colonies on the nutrient agar, and the rest of the 7 samples only showing only 1 type of colonies on the nutrient agar. And once the colony were grown on the culture medium, first we did the preliminary test, which included gram strain, catalase test, oxidase test, and hanging graph method. This gram strain was performed by Hucker's modification method with suitable controls. And here we could appreciate some of the collected organisms from our isolated samples. Here you could see the gram-positive cocci. Here you could see the gram-positive cocci and gram-positive bacilli in chains, and gram-positive bacilli, which is arranging in cuneiform shape, and gram-negative bacilli. This gram-negative bacilli only we further selected for the biochemical reactions to identify its genus level. And catalase tests were performed by slide method with suitable controls, and oxidase tests were performed by commercially available disc with suitable controls. And motility of an organism was seen by hanging graph method, and subcultured on enriched and differential media. Hello, ma'am. I hope I'm audible now.
Yes, you're audible. You're audible from the beginning. That text is.
Thank you.
It's not meaning to you. Okay, thank you. You can continue.
Thank you, ma'am. Thank you, ma'am. Thank you, ma'am. And subculture on enriched and differentiate media. Here, the blood agar were used for enriched media, and McConkey agar was used for differentiate media. In our blood agar, we isolated all the type of colonies like alpha hemolysis, beta hemolysis, and non-hemolysis, which is also known as gamma hemolysis, and dry wrinkled colonies. These are all the colonies were appreciated from our sample of fishmongers. And we also appreciated lactose fermenting or non-lactose fermenting from the McConkey agar. And biochemical reactions. All the gram-negative bacilli organisms were selected for doing these biochemical reactions, which included indole, TSI, which stands for triple sugar iron test, mental motility media. And tube coagulase staphylococcus from the coagulase negative staphylococcus, which is also known as cons. And results, gender distribution. Out of 50 fishmonger samples were taken out from the male, and the 15 samples were taken out from the female, which is estimated as 70% and 30%. On the other side, 10 samples from the male and the rest of the 10 samples were taken out from the female, which is estimated as 50-50%. And distribution of bacterial prevalence in fishmonger sand. Out of 50 samples, we isolated 55 types of colonies. Around the colonies, majority of the colony were identified as coagulase negative staphylococcus, which is estimated 30%. And the second frequently isolated organism were bacillus series, which is estimated as 20%. And Acinetobacter species were estimated as 12%, Escherichia coli were estimated as 10%, Klebsiella species were 9%, Staphylococcus aureus were 7%, Corynebacterium species were 6%, and the least frequently isolated organism were Aeromonas species, which is estimated as 2%. And distribution of bacterial prevalence in control group. From out of 20 control group samples, we isolated 34 colonies, and majority of the colonies were identified as coagulase negative Staphylococcus, which is estimated as 51%. And here also the second frequently isolated organism were Bacillus cereus, which is estimated as 25%. And Corynebacterium species were 18%, Staphylococcus aureus were 3%. And here the least frequently isolated organism were Acinetobacter species, which is estimated as 3%. And discussion. Fishmongers are considered to be high at risk of acquiring severe infections due to handling and processing of fish. Pathogenic organisms which can survive in any environment like water and soil can cause different clinical conditions to fishmongers. And there are some studies were already done on fishmongers and reported that Pseudomonas aeruginosa can cause unique skin manifestation to fishmongers, which is known as ecthyma gangrenosum. An Enterobacteriaceae family of Proteus vulgaris and Morganella morgani can cause skin cellulitis and urinary tract infections in fishmongers. This cellulitis can also be caused by Streptococcus ceneae, Vibrio vinificus, and Vibrio damselae. Erysipelothric rhusiopathy and Aeromonas hydrophilia can cause septicemia in fishmongers. In our study, we isolated higher amount of gram-negative bacilli in the hands of fishmongers. Even though they are asymptomatic, pathogenic organisms may also spread to other persons who cause infection. And some of the fishmongers were reported that they used to have urinary tract infection and skin infections for several times. This pathogenic organism may be responsible for causing that infections. So they need more effort to promote hygiene and hand washing behavior among them to control the infections among them to others. And all of this infection occur in both the immunocompetent and the immunosuppressed. Later, this will lead to increase the morbidity and mortality. And most importantly, fishmongers should be advised to wear gloves while handling the fishes, which will help to reduce the exposure to the pathogenic organism. And conclusion, the study concludes that the percentage of pathogenic bacteria was higher on the hands of fishmongers compared to the control group. Acinetobacter species, Escherichia coli, Klebsiella species are the most common pathogenic gram-negative organism isolated from the hands of fishmongers. The study helps to highlight the need for more intensive effort to promote the hygiene and hand washing behavior among them. And if hand hygiene behavior implement the important intervention like health education, then it will be the result in decreasing the risk of disease among them and others. And these are all the references I go through the study. And thank you so much for giving us this opportunity, ma'am. Thank you. Thank you, everyone. Hello. Ma'am?
Hello.
Ma'am, am I audible now?
Yes, yes, you are audible.
Yes, ma'am, I have finished my presentation, ma'am.
Okay. Session chairs, any questions for Mohan Kumar? Dr. Mukta, ma'am? Dr. Mukta, ma'am, do you have any questions for Mohan Kumar? Okay, thank you, Mohan Kumar. I hope our session chairs are having some technical issues. We are dealing with that. So moving on to the next presentation.
Thank you, ma'am. Thank you, everyone.
Yeah. Now we are moving with the next presentation by Joseph, oral presentation from Chettinad Academy of Research and Education on the topic biofilm detection among clinical isolates of Candida auris.
Ma'am,
Yes, yes. Sandipta, yes, you are having your presentation, right? You can go with the presentation before going to Joseph. Sandipta, you can, like wherever you stopped, you can go with the presentation from there.
Ma'am, I okay. Hello, ma'am. Can you see my screen? Hello, ma'am.
Yes, we can see you. We can see your screen.
So again, I started. Sorry, ma'am, there are noise come behind. Hello, ma'am, can you hear me now?
I can hear you. A small request to all the participants to mute their mics so that the session will remain undisturbed.
Okay, so sorry for the inconvenience. Now I am start the rest of my discussion. So how we can diagnosis and treatment. Diagnosis this nosocomial infection and how we treat it. Diagnosis. Many forms of nosocomial infection can be diagnosed through sight alone. Only we can see it and we can judge. We can define what is gradually it, which type of infection it. Like whose inflammation, rashes may be indication of infection. And blood and urine culture tests can be identified the infection so rapidly and very rapidly. And treatment. Now, how we can treat it? Treatment of nosocomial infection depend on infection type. If the infection occurs at the site of the catheter or other inserter line, the line should be removed immediately. Antibiotics can combat symptoms of many infections. A healthy diet, food intake, and rest can encourage natural healing process and prevent dehydration. Now, how we prevent nosocomial infection? We have to. There are nothing to prevent any bacterial contamination. We have to prevent it by some safety measures. So there are some safety strategies to prevent healthcare respiratory infection or nosocomial infection. First, it's a safety culture. Then, the following guideline, what is given by the hospital and nursing home and the other public health. And then, it's antibiotic stewardship. Then, it's surveillance. Then, it's screening and sorting patient and environmental hygiene. Then, it's hand hygiene. This is all about the nosocomial infection. And now, I would like to end my discussion. Thank you, everyone, and thank you all the professors present here and all everyone. Thank you. Hello, ma'am.
Yes, thank you for your presentation, Sandipta. Moving on to the Joseph M. presentation from Chettinad Academy of Research and Education on topic biofilm detection among clinical isolates of Candida auris.
Good afternoon, everyone. Ma'am, am I audible, ma'am?
Yes, you are audible, Joseph.
Okay, is my screen visible, ma'am?
Yes, it is visible. You can go with the presentation.
Good afternoon, everyone. Today we are going to see about our work in biofilm detection among the clinical isolates of Candida auris. First of all, you all may ask why I specifically selected Candida auris. Candida auris is a superfungus classified as critical in the World Fungal Priority Pathogens list of 2022. It poses an imminent threat to the persons with low immunity as it can cause fatal nosocomial infections. They are resistant to the common drugs which we prescribe for the normal fungus such as fluconazole, ketoconazole, amphotericin B, and intrinsically resistant to the echinocardians. Candida auris is commonly misidentified, and early detection is important in this treatment because the misidentification can lead to the wrong drug treatment. A biofilm is a microbial community of cell aggregates on the surface of under desirable conditions to form a thick layer capable of producing extracellular matrix, which is primarily composed of extracellular matrix, polysaccharides, deoxyribonuclease. Biofilm is produced when pathogen tends to attach itself to the moist surface by secrete a slimy glue-like substance. This is called as adhesion. A biofilm formation site can be in any place with solid liquid interface and this essential nutrients. It can also be formed at natural materials, metals, plastics, and also in the body and plant tissues. The formation of biofilm mainly takes place in 5 stages, which starts with attachment or adhesion. Then it is followed by colonization, proliferation, maturation, and final dispersion. This process repeats in various parts to produce further biofilm. As we can see in this image, the colonization of Candida auris is more in the skin than the mucous membrane, and the infections, those are mainly caused by the Candida auris, are mentioned on the right side of the image. Aims and objectives. Our aim is to assess the biofilm production among the clinical isolates of Candida auris. Objectives. Our first objective is to isolate the Candida auris from clinical specimen, then to detect the biofilm production using phenotypical methods, then to compare the results between the phenotypical methods, which are the Conger-Agar method, test tube method, and microdetector plate method. This study was conducted over 4 months in the mycology section under the Department of Microbiology, CHRI. A total of 450 isolates were included in the study, out of which 20 were identified in Candida auris, and 20 confirmed isolates from the archives that were isolated from the blood were also included. 3 tests were performed in triplicates to evaluate the biofilm production. Conger-Agar. Conger-Agar, this is a qualitative method for the detection of biofilm. The Conger-Agars were prepared by combining BHA broth, sucrose, and agar in 900 ml of distilled water. Anhydrous Congo red of 0.8 grams was dissolved in 100 ml of distilled water. Both were separately autoclaved and then mixed and then poured in the petri dishes. Inoculated isolates were incubated for 24 hours at 40 to 42 degrees Celsius and observed for the production of black colonies that indicates the biofilm production. As you can see in the image, the colonies which are being black are the one that produces the biofilm, whereas the red ones does not produce the biofilms. ATCC Candida albicans and ATCC Candida paraphylosis were used as control in this method. Test tube method. A standard Candida auris inoculum was added to 10 ml of SDB, which is saburo-dextrose broth, and incubated for 48 hours. After incubation, the broth was decanted carefully and the tubes were washed with PBS, phosphate buffered saline, and stained with 0.1% crystal violet for 15 minutes. The tubes were left to dry and observed for the biofilm production, which can be identified by the staining on the walls and in the bottom of the test tube. This is also a quantitative method for the detection of biofilm. And the microdetector plate method. This one is a qualitative method in which the above mentioned were the quantitative. Each 1 mcFarlane isolates were inoculated in the SDB broth, 1 ml per F1 drop, and was incubated for 24 hours. The wells were filled with 180 microliters of SDB and 20 microliters of the incubated suspension. It was covered and incubated for 24 hours at 37 degrees Celsius. The contents were decanted and washed carefully 3 times with PBS, which is phosphate buffered saline. The washed wells are stained with 0.1% of crystal violet and dried and read in ELISA reader at 450 nanometers and 650 nanometers. And the mean values were calculated. 450 isolates were included in the study, out of which 20 were identified in Candida auris. As said before, 20 confirmed isolates from the archives were also included. Candida auris are white to pale pink on high chrome agar and usually resistant to common antifungals, can grow well in 60% salinity and up to 42 degrees Celsius. They do not form pseudohyphae on Dalmas method. Growth on selective auris medium was also seen in all the 20 isolates. Among the 40 of the confirmed isolates that tested for the production of biofilm, 10 were positively produced the biofilms, which were given in the table 1 below. And following on to the tube method, that 10 of them were produced, as said before, biofilm. In those, 4 of them were strong, 4 of them were moderate, and 2 of them were weak. So totally 10. Following on to the microdetector plate method, this method is calculated by the optical density, and then mean values are taken. The mean values of the non and weak, moderate and high are given on the right side of the calculation. 4 of the clinical isolates were positive for heavy biofilm all production in 3 methods. It has produced black colonies with dry crystalline consistency in Conger-Agar and had shown strong production of biofilm in test tube method and has an optical density of 0.2634 in micrometers. 4 isolates were moderate producers and 2 were weak producers. Conclusion. We observed 25% of the biofilm producers, probably because of the 20 confirmed isolates from the archives, were from the patients of candidemia, which were mainly taken from the blood. Although unable to form biofilms equivalent to Candida albicans, Candida auris has a noteworthy virulence capacity that merits further studies. The simplest method to perform is growth on Congo red agar, which can be performed along with routine phenotypic tests to look for the biofilm producing isolates. This is valuable in patients with device-associated hospital-acquired infection. This method is simple, reproducible, and qualitative method. Results can be read up to 24 to 48 hours. Interpretation decontamination are automatically done within the limited time. These are the references which I took for this reference. Thank you, everyone. I would like to thank our HOD, Dr. Priyadarshini Shanmugam ma'am, who never said no to any researchers, and Dr. Ambujavalli ma'am, who have been with us in all the steps, and Abhita Evangeline, Ms. Abhita Evangeline, who is a research scholar, who helped me with all the procedures along the way. Thank you, ma'am. Hello, ma'am. Am I audible?
Yes. Thank you for your presentation.
Okay, ma'am.
Dr. Mukta, ma'am?
Yes.
Any questions for this presentation?
As I have disconnected from the meeting, so I have not listened the presentation properly. But can you please tell me the significance of your work?
Ma'am, my significance of my work is to mainly find out the biofilm production by Candida auris, ma'am.
Okay, biofilm production from?
Candida auris.
Candida auris.
Yes, ma'am.
Okay. How can you identify the Candida?
Ma'am, basically, we in a high chrome agar, it will be white to pink color colonies, ma'am. We will first do the gram staining, and after that, we will check them for the germ tube test, ma'am. It must be germ tube negative.
Test.
Yes, ma'am.
Okay.
If it is germ tube positive, then it will be Candida albicans, ma'am. After germ tube test, we will subject it to the salinity and temperature test, ma'am. It can grow on 16% of salinity and up to 42 degrees Celsius, ma'am. We had a separate agar named selective auris medium, which was specially meant for Candida auris, ma'am. It composed of for 100 ml of distilled water, agar 4 grams, peptone 2 grams, yeast extract 1 gram, glucose 2 grams, and NaCl 12.
Okay, fine, it's enough. Okay. Thank you so much.
Thank you.
Yeah, shall we go to other presentation, ma'am? Moving on to the other presentation by Dr. Hari Prasad, oral presentation from Chettinad Academy of Research and Education on the topic phenotypic detection of biofilms in Trichosporan species isolated from various clinical specimens. Dr. Hari Prasad.
Hello, ma'am. Can you see my screen? Is it visible?
Yes, we can.
Yes. So good afternoon to one and all gathered here. Good afternoon, ma'am. So before starting on with my topic, I wish to thank our HOD, Dr. Priyadarshini ma'am, for giving me this opportunity. And the study topic on which we are going to see is the phenotypic detection of biofilms in Trichosporan species isolated from various clinical specimens under the guidance of Dr. Ambujavalli. So let's go on into the study. Actually, Trichosporan is the second most frequently encountered opportunistic fungi, and this anamorphic yeast cell actually it causes from both mild infections like your white petra to severe fatal infections like your trichosporinosis in your immunocompromised individual, like patients with neutropenia or the patients with immunosuppressant drugs or patients with any malignancy. So this actual pathogenic activity of this organism is actually due to sessile cell communities, which is nothing but your biofilm, which actually clings to the host tissue and the medical devices. So the trichosporan itself actually encloses within this extracellular polymeric matrix, which facilitates it from escaping from the host immune response and the antifungal medication, which mainly leads to a therapeutic failure. This is actually a main concern in our clinical practice. You must also know that the biofilm is also going to act as a persister cell. So what is a persister cell? It's nothing but dormant cells which are actually responsible for relapsing infection once the antifungal medications are stopped. And we also know about the medically important trichosporan species like your Trichosporan asahi, Trichosporan inkin, and mucoides, which are actually showing a higher MIC, a minimal inhibitory concentration, and resistance to all your commonly used available antifungal drugs. So such a biofilm forming ability of this organism is actually of a medical importance. So biofilm detection is necessary for the treatment of this opportunistic infection. So my study, it mainly focuses on the isolation and the identification of the Trichosporan species from the various clinical specimens which we collected in our hospital, and the detection of biofilm using the following phenotypic methods. That is the Congo red agar, the test tube method, and the microtiter plate method. So the present study, which was actually carried out for a period of 1 year, we actually collected isolates from both outpatients and inpatients, which have actually been identified using the standard microbiological protocols. And the samples which we took included fungal keratitis, the urine samples, sputum, hair, stool, skin scrapings, vaginal swab, and the bronchoalveolar lavage. And all these samples were then screened using the Gram stain, where you can find out the arthroconidia and the blastoconidia cells in the Gram stain, and it also gives a urease positive. And this is the macroscopic appearance of the Trichosporan species in the SDA agar. For speciations, we then spent this samples formaldehyde off, under which we got about 100 Trichosporan species, in which 98 were Trichosporan asahi, 1 was Trichosporan inkin, and 1 was Trichosporan mucoides. So we have isolated and identified the Trichosporan species. The next method is detecting the biofilm. The first method is actually the microtiter plate method, in which we used a 96 well pre-sterilized polystyrene microplates to measure the biofilm. And then a suspension was actually made from the culture, which overnight Trichosporan culture on the SDA. And this suspension was inoculated into the wells. The quantity is actually 20 microliters of the suspension into the well, along with 180 microliters of the SDB, that is the saprodextrose broth, supplemented with 8% of glucose. Then we covered the micro plates and then incubated for 48 hours. And then these microplates were being washed thrice using the phosphate buffer solution. And then we added 1% of safran on the microtiter plates for 15 minutes, and it was sent for the ELISA reader. So my interpretation was actually with this faculty in our college, Vasanti and a HOD ma'am. This is the values which I took for my reference. Less than 0.120 were non-biofilm formers or a weak biofilm formation, and 0.120 to 0.240 were moderately forming, and more than 0.240 were high biofilm forming trichosporans. So the second method is actually the Congo red agar method. Ma'am, can you hear me?
Yes,
Hello?
we can hear you.
Yes.
We can hear you.
Yes, the second method is the Congo red agar method, which is actually the qualitative method for detecting the biofilm. The constituents used were the brain heart infusion broth, the sucrose, the agar number 1, and the Congo red indicator. All these were being used for making the Congo red agar medium. And according to the methodology, the following steps were being carried out. And then we inoculated the test organism into the Congo red agar plates, and it was being incubated for 24 hours. And black colonies with a dry crystalline consistency were found to be biofilm producing colonies, and while just the red colonies were the non-producers. And the final method is your test tube method, in which we took a loop full of organisms from the surface of the SDA plate, and these were being inoculated into the test tube, which contained about 10 ml of SDB, which was supplemented with glucose. Then we incubated it for 48 hours, and then after incubation, these the culture supernatant was being decanted, and the tubes were being washed thoroughly, and then we also stained it with 1% of crystal violet. So the excess stain was actually removed by washing it with a deionized water, and the tubes were being inverted, dried, and they were being absorbed for the biofilm formation. So coming on to the results from the total of 100 trichosporan species which we studied for biofilm formation, only 20 were forming biofilm. out of the microtiter plate method, we actually calculated, it showed adherent biofilm layers of optic density values ranging from 2.071 to 0.0162, in which 10 were forming mild, 7 were moderate, and 3 were heavily forming biofilm. And from the test tube method, we found that 3 were strongly visible biofilms which are adhering to the lining of the test tube wall. 5 were actually moderate and 1 was mild. Here you can see it, the strong, moderate, and the mild ones. And the third method is actually the Congo-Red-Aga method, in which actually we got the 20. Out of those 20, 17 actually formed black colonies with a metallic sheen, while the other 3 was just forming a black discolorization of the media. So with this, we'll come into the results and discussion. So actually, this study was conducted for a period of 1 year after the IHEC approval from our college, from Mycology Section, Department of Microbiology, CHRI. All the trichosporon isolates were actually from the clinical samples which we collected in our hospital. They were being studied for biofilm production. Out of the 100 trichosporon, we found 20 were producing biofilm. And in our study, we also found that majority of the trichosporon asahi were being producing the biofilms. So the methods I used were the Congo-Red-Aga, the microtiter plate method, and the test tube method. So out of these 3, I found that the microtiter plate method was the best comparatively the others, and the Congo-Red-Aga was also used as a screening medium prior to perform the microtiter plate method. So in conclusion, I wish to say that the biofilm is one of the important virulence factor of trichosporon species, and the early detection of the biofilm production using the Congo-Red-Aga, the microtiter plate method may be useful in preventing therapeutic failure, the opportunity for the gene transfer between the organisms, and may also apply as a clinical decision tool before we administer antifungal drugs, because most of the immunocompromised patients are resistant to all these antifungal medications. That's it. So these were my reference. So thank you.
Yeah, thank you for your presentation, Hari Prashant.
Yes, ma'am. Thank you, ma'am.
Mukta ma'am, any questions for him?
Dr. Hari Prasad,
yes,
can you tell me what type of disease are caused by the trichosporum?
Ma'am, it can cause superficial infections like a white pedra, but most severely it causes an immunocompromised patient. It causes trichosporinosis, like disseminate. It is an acute febrile conditions, like a disseminated condition. It mainly attracts towards the renal, ma'am. It can cause urinary tract infections, and it can cause a renal failure. A renal failure. Yes, ma'am.
One more simple question. You have mentioned that trichosporum is a opportunistic fungus. So what do you mean by the opportunistic word?
Opportunistic, ma'am.
Yes.
Opportunistic is nothing but it is a actually it is present normally in all human beings. We are we have a stable immune system. Only in people with example HIV or the patient with neutropenia, all these fungi take over the host immune response, and it causes an opportunistic infection. For example, we can also take your mucormycosis, which we found it in your COVID patients.
You mean generally it is a non-pathogenic, but sometime when it may get an opportunity that then it may be pathogenic. Right?
Yes, ma'am. Yes, ma'am. And the reason for me getting a very low biofilm is we used different samples. For example, fungal keratitis, sputum, all those. If we have specially stick on to the urine or blood, we would have got a more biofilm forming trichosporons.
Okay. Very good presentation.
Yes, ma'am.
Thank you so much.
Thank you so much, ma'am. Thank you.
So moving on to the next presentation by Vandana Rani, poster presentation from Lovely Professional University on the topic plasmid mediated linezolid resistance among Heterococcus faecium, phenotypic characterization and molecular epidemiology. Vandana Rani, are you there?
Yes, ma'am. I am audible, ma'am.
Yeah, you are audible. You can start sharing your screen.
Okay, ma'am. Okay. Ma'am, my screen is visible.
Yes, yes, your screen is visible. Could you
Okay.
please zoom
Okay,
in?
okay.
Zoom in. It is not clear. Could you please make some zoom?
Okay, okay, ma'am. I will. Okay. Now it is.
Yes, now it is clear.
Okay, ma'am. Okay. Hi, good afternoon, everyone. Myself, Vanna. I am pursuing my PhD under the supervision of Dr. Amin from Lovely Professional University, and I am carried out my work in the Safdarjung Hospital under the supervision of my co-supervisor, Dr. Rajni King. Enterococcus faecium is a gram-positive bacteria, is the third most common cause of nosocomial infection, and vancomycin resistance has emerged in Enterococcus faecium. WHO World Health Organization has classified vancomycin-resistant Enterococcus faecium as a high-priority pathogen, and linezolid is the reserve antibiotic for vancomycin-resistant isolates. Linezolid resistance is commonly mediated by a mutation in 23 sRNA, plasmid-mediated gene by CFR and OPTRA gene, and limited study have investigated the association of OPTRA gene with linezolid resistance from India. The aim of the study was to determine the antimicrobial susceptibility and the prevalence of plasmid-mediated resistance among linezolid-resistant E. faecium isolate, and phenotypic characterization and molecular epidemiology of linezolid-resistant E. faecium isolate was also studied in this study. The material and methodology. Clinically significant linezolid-resistant E. faecium isolate were enrolled in the study. The identification was done by the Vitek 2 automated system, which is used for the identification of bacterial isolate and fungal isolate too. Antimicrobial susceptibility was tested by the diffusion method, and the MIC for vancomycin and linezolid was determined by the E-test. And for the detection of resistant mechanisms like OPDR gene and CFR gene, PCR was performed, and genetic relatedness in among linezolid resistant E. faecium isolate was done by the pulse field gel electrophoresis. The primer sequence and their amplicon sizes are given in this table, which are used in the study. And DNA extraction was extracted by the boiling prep method by the MD Elgin and LC Kelly. Here are the results of the study. A total of 30 clinically isolate from the patient with nosocomial infection of urinary tract infection and the sepsis were enrolled. The MIC of linezolid was more than or equal to 8 microgram per ml for the or isolate. All isolate were MDR, and the XDR was observed in the 93% isolate. Majority of the patient had the comorbid condition. All isolate were found negative for CFR gene, and the prevalence was for the OPTRA gene was very high in our state, which was the 80%. And the pulse field gel electrophoresis analysis demonstrate that the three major clone among the linezolid resistant E. faecium isolates. The antimicrobial profiles associated pattern are given in the table, like the 100% isolate. As I said earlier, the 100% isolate were the MDR. Just here you can see that the ampicillin resistant was observed in the all 30 isolate. Ciprofloxacin resistant was also seen in the all 30 isolate. Like only 3% isolate was susceptible to the high level gentamicin, and 5% for the nitrofurantoin, 5% chlorophenicol, and only 3% isolate were observed susceptible to the tetracycline. Here you can see an image of the antimicrobial susceptibility pattern. Figure image 1 showing the ATCC isolate 29212 isolate, which is, here you can see this isolate is susceptible to the all antibiotic according to the CLSI guidelines. And here you can see the clinical isolate, which is a linezolid resistant isolate, and here you can clearly see that this isolate is the resistant to the all antibiotic. And in figure 2, you can see the MIC of linezolid. In figure A, the you can see the MIC of a susceptible isolate, which is a ATCC 29212, and in figure B, you can see the MIC of by e-test of a lineage resistant isolate. Here you can see there is no zone of clearance around the e-test. Now come to in this figure, you can see the amplification of the OPT PCR amplified product of OPT RAG, which is 1395 base pair, and in this figure you can see the amplification of the CFR gene, which is a 746 base pair. Here you can see the dendrogram generated by the generated for the pulse field gel electrophoresis, and we got the three major clone A1, A2, and A3. Those are the genetically related. Here are the conclusions. This is the first report of OPTR mediated lineage resistant in Anthropogus faecium from India. All infection were the nosocomial. The study suggests that the OPTR is the important mechanism of lineage resistant, as OPTR gene is frequently associated with the plasmid, and it can be transferred by the transfer from the susceptible gene to the susceptible isolate to the it can be transferred from the resistance isolate to the susceptible isolate by transferable resistance. Implementation of infection, here we can conclude that the implementation of infection control practices are important to prevent further spread of lineage resistant in our hospital. Thank you. Here are some references.
Thank you for your presentation, Vandana. Mukta ma'am, any questions for her?
Vandana, what do you mean by the MIC?
Minimum inhibitory concentration, ma'am.
Can you define this?
Yes, ma'am. What is the minimum concentration is required to inhibit the bacterial bacteria growth?
Okay. Is there any difference between the MIC and MBC?
Yes, ma'am.
What is that? MBC is minimum bactericidal concentration, and MIC is minimum inhibitory concentration.
Inhibitory concentration.
Can you differentiate the both? Can you differentiate?
Ma'am, MIC demonstrate the lowest level of antimicrobial agent to inhibit the growth of bacteria or any pathogen.
Okay.
And MBC is the lowest level of antimicrobial agent, resulting in, I think, antimicrobial agent, microbial death. Sorry.
Microbial
death. मतलब is MIC means it's inhibit the growth, and MBC it's completely
kill the growth, है ना? Bactericidal.
Yes, ma'am. Yes, ma'am. Bactericidal. It's completely kill the bacteria.
Okay, fine. Can you differentiate the phenotypic and genotypic characterization?
For ma'am.
Can you differentiate between the genotypic and phenotypic characterization?
Phenotypic characterization was based on the biochemical test. Like for Enterococcus faecium, we will use the catalase test, bilesculin, and the growth on the high salt concentration like 6.5 NaCl. And for genotypic characterization, we will do the 16S RNA sequencing for the species identification. And for for the specific resistance mechanisms, we can do the for lineage resistant, we can do the 23S RNA mutation sequencing and OPTR gene and CFR gene.
Okay. Very good presentation. Thank you so much, Vanna.
Okay, ma'am. Thank you.
Is there any presentation left?
Yes, ma'am. We are left with 1, 2, 3, 4, ma'am. Out of 4, 2 are poster presentations and 2 are video presentations.
Okay.
Now we are going with the presentation of Kavana CP, a poster presentation. She is from JSS Academy of Higher Education and Research. She's gonna give her presentation on the topic, evaluate the efficiency of MIR1 virulent protein as a vaccine candidate against acute toxoplasmosis in BALC mice.
Hello, ma'am.
Hello.
Can you see my slides, ma'am?
Yes, I can see your slides.
Okay, ma'am. Good afternoon, everyone. My name is Kavana CP. Today, I'm going to present a poster presentation on evaluate the efficacy of MYR1 virulent protein as a vaccine candidate against the acute toxoplasmosis in BALC mice. Coming to the introduction, Toxoplasma gondii is a zoonotic disease caused by a single cell parasite that can infect the human and other animals, and it is particularly concerning for pregnant women due to the risk of the congenital disability or the miscarriages. Unfortunately, there is currently no effective vaccine available to prevent the T. gondii infection. However, our current study aims to develop an early vaccine that could reduce the severity of the diseases and protect against this dangerous pathogen. Coming to the need for the study, early vaccination with the MYR1 protein in the T. gondii can provide a pre-exported protection against the infection, reducing the risk of the survived health sequences in the high-risk population, and targeting the multiple epitope within the MYR1 can induce a robust and broad immune responses, improving the protection against the parasite and reducing the risk of the antigenic variation and escape. Effective vaccination against the T. gondii could have significant economic benefit by reducing the cost associated with the treatment and management of the toxoplasmosis. And early vaccination approaches using the MYR1 protein in the T. gondii as a potential to prevent and control the spread of the parasite and improve the public health outcome. Coming to the material and methods, in our study, we developed a multi-epitope vaccine against the T. gondii by targeting the MYR1 protein. We started by designing the vaccine using an immunoinformatic approaches, followed by cloning it into the EGFP vector and confirming the transcription and translation via PCR. We then construct immunization followed by the cytotoxic analysis. Next, we test the different doses of the vaccine were treated to the BALC mice in order to evaluate the efficacy of the vaccine. Coming to the result and discussion, our first step was to construct the cloning using the EGFP vector with the green part representing the gene coding of the vaccine and the black indicating the vector backbone. We successfully amplify the MYR1 protein by the PCR and ligated into the TGB MYR1 plasmid. We then constructed the we then conducted the Western blotting analysis to predict the mass of the T. gondii in the HE3 cell transformed with the TGB MYR1, and we obtained the 0.5 mg yield from the clone. The synthesized vaccine was further evaluated for the cytotoxicity studies on the HFF cell lines. Further, we estimate overall survival rate of the BALC mice after vaccination. Compared to the standard vaccine, our designed vaccine shows the more survival rate. We shows the potency of our vaccine. Further, in order to check the morphological and behavioral changes. Coming to the summary and the conclusion, in summary, the study evaluated the efficacy of the MYR1 virulent protein as a vaccine candidate against the acute toxoplasmosis in the BAC mice. The result demonstrated that the MYR1 protein induces a significant immune response and provide a partial protection against the parasite infection. In conclusion, the MYR1 protein holds the promise as a potential vaccine candidate against the acute toxoplasmosis, although further studies are needed to optimize its effectiveness and evaluate its safety for uses in the human. At last, I acknowledge special thanks for the ICCAM and also the biotechnology and the bioinformatics, JSS Academy of Higher Education and Research, Mysuru, for giving us the opportunity to present the work. Thank you, ma'am.
Yeah, thank you for your presentation. Ma'am, it's over to you for question and answers.
Can you tell me how your research is beneficial for the society?
Ma'am, Toxoplasma gondii is the zoonotic diseases, ma'am. It transfers from animal to the human, and it mainly targeted for pregnant women, and it also causes the neurological diseases. For giving the early vaccination, we can stop the Toxoplasma gondii spread. So it helps the it helps to improve the we really want to stop the spreading of the Toxoplasma gondii in the early stage only.
Is there any vaccine available for this disease?
Ma'am, for the animal, one vaccine is available. For human, there is no vaccine. For sheep, it is one vaccine available.
Okay. So you are trying to find out the vaccine against this disease.
Yes, ma'am.
Okay, very good. Thank you so much.
Thank you, ma'am.
Excuse me, ma'am. Hello. Ma'am, shall we go for other presentation? Hello, am I audible?
Yes, ma'am.
Okay, moving on to the other presentation by Aishwarya H. from JSS Academy of Higher Education and Research. In the topic, evaluating the potential of USP-A1 protein as a promising vaccine candidate for Moraxella catarrhalis. Aishwarya, are you available?
Yes, ma'am.
Okay, you can start sharing your screen and go with your presentation.
Okay, ma'am. Ma'am, my poster is visible.
Yes, your poster is visible, but it is not clear. It is completely blurred.
Yes, ma'am.
I can see your poster, but it is not at all clear. Could you please zoom in?
Okay, ma'am. Is it fine, ma'am?
Yes, now it is fine. You can continue.
Okay, ma'am. Good afternoon, everyone. Good afternoon, everyone.
Yeah, you are audible. You can continue with your presentation.
Good afternoon, everyone. Myself, Aishwarya. I'm from JSS Academy of Higher Education and Research. Today, I'm going to present a poster presentation on evaluating the potential of USP-A1 protein as a promising vaccine candidate for Moraxilla catarrhalis. Introduction. Moraxilla catarrhalis is a gram-negative bacteria that commonly colonizes in the upper respiratory tract of humans. It causes middle ear infection in children, and it is a main cause. It is a prominent bacteria for the cause of COPD. And it has a significant disease burden associated with Moraxilla. There is no currently licensed multi-epitope vaccine available for this bacteria, and the severance of the bacteria is high. In Moraxilla, USP-A1 protein is a well-characterized surface antigen that has been shown a strong immune response in animal model, in human study, and in human studies. USP-A1 plays a crucial role in the pathogenesis of Moraxilla catarrhalis. USP-1 enables the bacteria to adhere and invade the host cell by binding to the various host receptors. These receptors include transferrin and lactoferrins. These receptors allows the bacteria to adhere to human cells. And USP-A1 helps the bacteria to avoid human host immune defense and establish infection in humans and in upper respiratory tract. There is a need for the studies. Moraxilla is a highly drug resistance bacteria which is present. There are few available drugs which are used for the treatment of Moraxilla, but they have a highly adverse effect on human body, and bacteria has been showing a adverse antibiotic resistance for the existing drugs. So there is a need for the study for the development of a vaccine for this bacteria to avoid the upper respiratory tract infection.
Hello.
1 minute, ma'am. Let's move on to the materials and methods. First, we retrieve the targeted sequence, and then we built a homology modeling construction. Then we predicted the secondary structure of the protein. Further, we predicted the T cells and B cells after screening the T cells and B cells. Then we constructed the multi-epitope vaccine. After the construction of the vaccine, we went for the structural validation of the vaccine. Immunosimulation was the method used for the structural validation, and to see the stability of the vaccine, we used immunosimulation. Then we did the cloning by using the E. coli vector. Results and discussion. In a figure 2, you can see the multi-epitope sequence of the USPA1 protein, and this is the multi-epitope structure of the protein. These are my results for the immunoinformatics results and simulation results. This is the cloned E. coli vector for our vaccine, and in silico restriction cloning of the gene of the vaccine constructed into a MAPLE vector. The red indicates the gene encode the multi-epitope vaccine, and the black part indicates the vector backbone. And these are my docking results. Like first we got the docking results, promising results. Then we moved to the structural validation for the MD simulations. My sincere acknowledgement summary and conclusion. There is a lack of studies and preventing measures for Moraxilla, and there are significantly associated with respiratory tract infection. This pathogen has been named during the time of COVID, and it has been playing a significant role in upper respiratory tract infections. So there was a need for a need to prevent this bacteria before it causes the upper respiratory tract infection. There was no multi-epitope vaccine available, so we are by using the in silico studies, we are building a vaccine for the Moraxilla. And our constructed vaccine showed a our vaccine showed sorry for the disturbance, ma'am. Our vaccine showed a good results in the simulation and in the structural validation. My sincere acknowledgement to the authors and the ICCMA, and I also like to thank JSS AHER, Department of Microbiology and Biotechnology, for carrying out my work. Thank you, ma'am.
Thank you, Aishwarya. Mukta ma'am, now it's over to you for the questions. I hope ma'am was not available here due to technical issues. Okay. Aishwarya, you can stop sharing your screen.
Thank you.
Now we are moving with our video presentation. Video presentation by K. Vijay Anand from Meenakshi Academy of Higher Education and Research on the topic effectiveness of music therapy on stress, anxiety, and depression among clients scheduled to undergo coronary angiography.
The topic is effectiveness of music therapy on the stress. A warm welcome to everyone for this presentation. The topic is
participants, can you see my screen? Participants, can you see the video?
A warm welcome to everyone for this presentation. The topic is effectiveness of music therapy on the stress, anxiety, and depression among clients scheduled to undergo coronary angiography. The stress is an unpleasant psychological and physiological state caused due to some internal and/or external demands that go beyond our capacity. The activation of hypothalamic pituitary adrenal axis and sympathetic nervous system due to The depression is often associated with a variety of medical conditions, particularly endocrine disorders, cardiovascular diseases, neurological disorders, autoimmune disorders, and malignancies. It is common for a person to feel sad or depressed after a myocardial infarction, before a cardiac surgery or procedure, recent hospitalization, or a new diagnosis of heart disease. So undergoing invasive procedures such as coronary angiography may be acutely stressful for the clients. The factors that contribute to the client stress include pain, unfamiliar environment, and fear about the outcome of procedure. The coronary heart diseases ranges from 1.6% to 7.4% in rural population and 1 to 13.2% in urban population. And age standardized coronary heart disease death rates in people between 30 to 69 years are 180 per 1 lakh population in Britain, 280 per 1 lakh population in China, and 405 per 1 lakh population in India, which is very high. And 50% of coronary heart disease related deaths in India occur in people less than 70 years of age. The need for the study: the researcher observed that when the patients are relatively posted for coronary angiography, they become stressed, anxious, and having negative thoughts and depression about the outcome of the procedure as well as about the future. The physical symptoms of anxiety and stress such as tachycardia, palpitations, tachypnea, and increased blood pressure further increases the workload of the ailing heart, which will hinder the prognosis of the client. So many research found that music therapy has beneficial effects in reducing the stress and anxiety of the clients in a variety of settings, and it is also inexpensive. The objectives of the study are to assess the level of stress, anxiety, and depression among clients scheduled to undergo coronary angiography, to evaluate the effectiveness of music therapy on stress, anxiety, depression among clients scheduled to undergo coronary angiography, to compare the effectiveness of music therapy on stress, anxiety, depression between the experiment and control group clients, and to associate the stress, anxiety, and depression with selected demographic variables of experimental group clients, and to correlate the relationship between stress, anxiety, depression in experimental group and in control group clients who are scheduled to undergo coronary angiography. The hypothesis: there will be a significant reduction in the level of stress, anxiety, and depression between the pre and post intervention with music therapy among clients who are scheduled to undergo coronary angiography. And there will be significant reduction in the post-test levels of stress, anxiety, and depression between the experiment and control group clients who are scheduled to undergo coronary angiography. The research design is a randomized control trial, and the experiment group were asked to listen 30 minutes of classical Carnatic instrument violin music based on Todi Raga through headphones, and control group clients were not provided music therapy but asked to take complete bed rest without any interruption for 30 minutes. The variables are independent variable, music therapy, dependent variable, stress, anxiety, and depression. The setting of the study, it was conducted at Intensive Coronary Care Unit, Department of Cardiology, Government Rajaji Hospital, Madurai. Sample. The study sample comprised of clients who are scheduled to undergo coronary angiography and who fulfilled the criteria for sample selection. The sampling technique. The subjects were selected using simple random sampling lottery method based on the criteria for sample selection. The sample size is 60. So 30 subjects are assigned in experimental group and 30 were in control group. The criteria for sample selection, inclusion criteria: patients who are willing to participate, patients who are relatively posted for coronary angiography, patients who can read or write or understand either Tamil or English. And both male and female clients will be included in the study. And exclusion criteria: patients who are having hard of hearing, patients who are taken for emergency coronary angiography, patients who had already undergone coronary angiography, patients who did not have stress, anxiety, and depression. Description of tool. So section A contains the demographic questionnaire. So as age in years, gender, religion, educational status, etc. Section B is depression, anxiety, stress scale. That does 21 item scale to assess the level of stress, anxiety, and depression. And section C, assessment of biophysiological profiles like patient's pulse rate, respiratory rate, blood pressure, and peripheral oxygen saturation, SpO2. The procedure for data collection. So pre-assessment was done to assess the level of stress, and biophysiological profile was also recorded. And a simple random lottery method was used to assign the patient in experimental and control group if they satisfy the inclusion criteria. And subjects in the experimental group are asked to lie comfortably and were instructed to listen to the 30 minutes of music recording, violin music based on Todi Raga. And the subjects in the control group were asked to take uninterrupted bed rest for 30 minutes by lying down comfortably, and asked to avoid reading, speaking on telephone or listening to radio, etc. And after 30 minutes, post-test assessment was done for the subjects in both groups. This is the comparison of mean and standard deviation between pre-test and post-test measurement of stress, anxiety, depression, and biophysiological profile of clients in the control group. There is no statistical significantly association found except for pulse and respiration. Both are statistically significant. That may be due to taking complete bed rest for 30 minutes without any interruption or any disturbance from outside. This is the comparison of mean and standard deviation between pre-test and post-test measurement of stress, anxiety, depression, and biophysiological measures in experimental group. In this, almost every variable that are found to be statistically significant. The stress, anxiety scores, depression scores, pulse, respiration, SpO2, systolic BP, diastolic BP. The mean difference is good, and the p-value is also statistically significant. This is the graphical representation. The unpaired t-test for evaluating the post-test effectiveness of music therapy on stress, anxiety, and depression between experimental and control group. So there is a statistically significant improvement has been found in the experiment post-test controlled post-test with the stress, the anxiety, and depression. T-test values are 8.398 for stress. The p-value is 0.000. It is highly significant. And anxiety t-test value is 8.125, which is also statistically highly significant. Its p-value is 0.000, and depression is also statistically highly significant with the value of t-value of 3.181 and a p-value of 0.000. The major findings of the study: the mean stress score in the experimental group reduced from 27.52 to 12.33 in post-test. that was statistically significant with pair t-test value of 10.57 and p-value of less than 0.001. The mean anxiety score in experimental group reduced from the pre-test score of 18.8 to 7. The pair t-test result shows statistically significant reduction in the level of anxiety, that is t-score 9.088 and p-value of less than 0.001. The mean depression score reduced from 17.87 to 25.2 after music therapy, and which was highly significant with t-test value of 8.48 and p-value of less than 0.001. In experimental group, all the parameters in the biophysiological assessment is reduced and they were statistically significant. A significant association was noted between the gender and the level of stress among the clients belong to post-test experimental group. The chi-square value of 4.509 and p-value of 0.03 with 1 degree of freedom. The conclusion. The results of the study showed that the clients who listened to 30 minutes of soft melodious violin music based on Todi Raga has a statistically significant reduction in the levels of stress, anxiety, and depression before going coronary angiography than those in the control group. And the music therapy was cost-effective, non-invasive, and free from side effects and highly feasible. And it can be used as an alternative and effective intervention to reduce the stress, anxiety, depression for those who are undergoing coronary angiography. Thank you for the opportunity.
We are having one more video presentation. I'm going to share that.
A warm welcome to one and all for my presentation today. The topic is a study to assess the severity of alcohol dependence among male clients attending psychiatric outpatient department. The objectives of the study are to assess the alcohol dependence among male clients attending psychiatric outpatient department at Government Rajaji Hospital, Madurai, and to analyze the demographic variables of alcohol-dependent male clients attending psychiatric outpatient department, and to associate the selected demographic variables with the severity of alcohol-dependent syndrome. The hypothesis, there will be a significant association between the selected demographic variables and the severity of alcohol dependence. The research design, descriptive study design was used in this study, and setting of the study, it was conducted at the psychiatric outpatient department at Government Rajaji Hospital, Madurai. The population, the target population are all the clients who are attending psychiatric outpatient department for the treatment of alcohol dependence. And the accessible population is clients attending psychiatric outpatient department for the treatment of alcohol dependence at Government Rajaji Hospital, Madurai. Sampling technique, convenient sampling technique based on the inclusion criteria was used in this study. And sample size is 30 clients who fulfill the inclusion criteria. The inclusion criteria are The male clients who are aged 20 years and above, and clients who are willing to participate in this study, and clients who are able to understand, speak either Tamil or English. Exclusion criteria are clients who are having sensory deficit, clients who are delirious and acutely ill. The description of tool. The tool for data collection consists of two sections. One is demographic data that contains basic details such as age, occupation, income, family type, etc. And section B is severity of alcohol dependence questionnaire, SADQ. It consists of 20 items. Each item is scored on a 4-point scale. The descriptives are almost never to nearly always. That results in a corresponding score of 0 to 3. So the total score ranges from 0 to 60. Score of 31 or higher indicates severe alcohol dependence. A score of 16 to 30 indicates moderate dependence, and score of below 16 usually indicates a mild physical dependence. The method of data collection: after obtaining a formal permission from the hospital authorities and based on the inclusion criteria, by using convenience sampling technique, 30 clients were selected and initial rapport was established. And the purpose of the study was explained to the clients. And the structure interview method was used to collect demographic variables as well as alcohol dependence severity questionnaire, SADQ. The data analysis are done by descriptive and inferential statistics. And this is the percentage distribution of clients according to their age. So most of the clients are aged between 20 to 29 years. That contributes 34%. And this is the percentage distribution of clients according to the type of family. So 73% of patients are from nuclear family. And this is the percentage distribution of clients according to their occupation. So 30% of clients are from daily wages group. And this is the severity of alcohol dependence in respect of selected demographic variable. In this, age in years has a significant association with the severity of alcohol dependence. That is the calculated chi-square value 6.33. And that is more than the tabulated value of 5.99 at the significant level of 0.05. So apart from that, religion, education, occupation has no significant association with the severity of alcohol dependence. And the income, nuclear family, marital status also has no statistically significant association with the severity of alcohol dependence. But the duration of consumption has statistically significant association with the severity of alcohol dependence. The calculated chi-square value is 9.31, which is more than the tabulated value of 7.82. That is significant at the level of 0.05. This is the graphical representation according to the severity of alcohol dependence with their age. This is the severity of alcohol dependence with the years of consumption. The conclusion arrived in the findings are the majority of subjects belong to the age group of 20 to 29 years, and majority of alcohol dependence clients were married and from nuclear family. The severity of alcohol dependence increased with the age and duration of alcohol consumption, which are statistically significant. The clients from nuclear families had severe alcohol dependence compared to the clients from joint families. Thank you for the opportunity.
So that's about the video. We have one last presentation of video. So I'm gonna play that.
Morning to all. Today I am going to present my study, a comparative study to assess the magnitude of corporal punishment between attention deficit hyperactivity disorder and normal children. Introduction. Today children are tomorrow's citizens. As a professional of pediatric healthcare, it is our duty to make this vision a reality, and achieving this goal involves a coordinated and dedicated approach from different scientific disciplines, requiring efficient and enormous effect. The future of our country depends on the child health and strength of our young people. However, many children have mental health problems that interfere with normal development functioning. Attention deficit hyperactivity disorder is a behavioral disorder characterized by persistent inattention, hyperactivity, and impulsivity that impairs educational achievement and social functioning. It is diagnosed by ascertaining whether the child behavior meet the specific criteria enumerated in the Diagnostic and Statistical Manual of Mental Disorder. The pediatrician and the nurses can play an important role in detecting this condition early, instituting appropriate treatment, counseling the parent and the class teacher, and monitoring the child academic and psychosocial progress on a long-term basis. Most of the ADHD children are getting punished by the parent and family member and other significant people in the lives infrequently because of their poor performance, disruptive and socially embarrassing behavior. Research shows, in adding to the proper pharmacotherapy, effective psychosocial and behavioral management are also beneficial in children with ADHD. Objective of the study. First one is to assess the magnitude of corporal punishment in ADHD and normal children. To compare the magnitude of corporal punishment of ADHD and normal children, to associate with selected demographic variable of ADHD and there will be a significant difference in the magnitude of corporal punishment meted out to attention deficit hyperactive disorder and normal children. Delimitation. The studies was limited to 6 weeks. Data collection procedure through the interview technique. Methodology. Research design. The comparative descriptive design examine and describe difference in variable 2 or more groups that occur naturally in the society. Setting up the study. The study was conducted at Child Guidance Clinic for Attention Deficit Hyperactive Disorder Children, Immunization Clinic at Institute of Child Health and Hospital for Children, Egmore, Chennai 8. Study population, study group. The children aged 4 to 12 years and diagnosed to have attention deficit hyperactive disorder in Child Guidance Clinic at Institute of Child Health and Hospital for Children, Egmore, Chennai 8. Normal group. The normal children aged 4 to 12 years who were attending immunization clinic at Institute of Child Health, the Hospital for Children, Egmore, Chennai 8. Sample size, study group. A sample of 50 attention deficit hyperactivity disorder children who were attending Child Guidance Clinic at Institute of Child Health and Hospital for Children, Egmore, Chennai 8. Normal group. A sample of 50 normal children who were attending the immunization clinic at Institute of Child Health and Hospital for Children, Egmore, Chennai 8. Sampling technique. Convenient sampling technique was adopted for this study. According to Pollitt and Agar 2009, convenient sampling proceeded on the belief of that researcher knowledge both the population and its elements can be used to add pick the cases included in the sample. Criteria for selection of the sample. Inclusion criteria. Study group. Children aged from 4 to 12 years with attention deficit hyperactive disorder, both boys and girls. Children who are attending the child guidance clinic at Institute of Child Health and Hospital for Children, EGMO, Chennai 8. Mothers who are speaking Tamil and English. Normal group. Children aged from 4 to 12 years, both boys and girls. Children who are attending immunization clinic at Institute of Child Health and Hospital for Children. Mother who are speaking Tamil and English. Analysis and interpretation. Percentage of different aspects of corporal punishment in among normal children. The above table show the mean score and percentage score of the different aspect of corporal punishment among normal children, in which punishment score was mean is equal to 1.18% and score is equal to 23.6%. And reason was mean is equal to 2.10 and percentage score is equal to 52.5%. Mode of punishment was mean is equal to 1.12, percentage score is equal to 28%. And sequel was M is equal to 1.74 and percentage score 43.5%. And severity of injury was mean is equal to 0.46 and percentage score is equal to 15.3%. The overall mean score was 6.60 and the percentage score was 32.58%. In this study shows the overall mean was 16.34 and standard deviation was 2.14 for ADHD children, whereas the normal children mean was 6.60 and the SD was 1.72 and they statistically found to be significant to 23.33, P is equal to 0.001. Hence, the research hypothesis there will be significant difference in the magnitude of corporal punishment. Mothers meted out to attention deficit hyperactive disorder normal children was accepted. The study result represent the level of corporal punishment among 50 mothers of ADHD children, in which 76% of mothers of attention deficit hyperactive disorder children were giving moderate corporal punishment, 24% of them giving severe punishment with regard to normal children, 90% of mothers were giving moderate punishment, and 10% were giving mild corporal punishment. Conclusion. ADHD children 4 to 12 years of children is a common condition, and the mothers play an important role to identify the need of their children and guide so that they can modify the problem of children. The study revealed that ADHD children were more often punished by various reasons and they suffer severe injury when compared with normal children. They also poorly responded to punishment as comparing with normal children. From this study, it may be concluded that the magnitude of corporal punishment was high practiced by mothers among attention deficit hyperactive disorder children than the normal children. Thanking you.
Hi, if Ms. Deepika, you're ready for your presentation, please go ahead.
Yes, ma'am.
Please share your screen and you can start. After that, Ms. Preeti, if you're available, you would have the presentation and we'll wrap up with that. Thank you.
Ma'am, can I present?
Yes, yes, please. Please go ahead.
Okay.
Deepika, can you start your presentation? Ms. Deepika, are you available? Okay, if Ms. Deepika is not available at the moment, we can go ahead with Preeti if you're present, or we will have to wrap it up.
Yes, ma'am.
Preeti, okay, if you're available, please share your screen and start your presentation.
Okay, ma'am. Okay.
Thank you.
Ma'am, is my screen visible? Ma'am, is my screen visible?
We can see thing says Preeti Man has started screen sharing, but your PPT is not visible yet.
Am I audible, ma'am, now?
Yes, you're audible. We cannot just see your screen. It's just starting.
Now, ma'am, is my screen visible now?
Not yet, Preeti. Yeah, we can see it now. Yes, okay. Please start the presentation. We can see your screen. Yeah.
Okay, ma'am. Okay. A very
Thank
healthy
you.
and warm good afternoon to all present here. I'm the student of SGT University from MSc Microbiology, going to give the presentation on my own topic, rhizobacterial secondary metabolites as a stress amylotic agent in plants. So the first is introduction. So the first question arises is, what is secondary metabolites? They are such as osmoproteins, volatile organ compounds, and exopolysaccharide are one of the most diverse and fascinating classes of phytochemicals that accumulate in rhizosphere and plant tissues for a variety of functions. Ma'am, am I audible?
Yes, you're audible.
Okay, ma'am. Okay.
Yeah, you can go ahead. If you're not audible, I'll tell you. Okay, so you can just keep up.
Okay, ma'am. Okay. Osmoproteins include soluble sugars, proline, and polyamine. And volatile organ compounds include aldehydes, ketones, and alcohols, plant hormones. Plant hormones like abscisic acid, cytokines, and ethylene. So, SM aid a host in important functions such as protection, competition, and species interactions. As I mentioned in introduction, the primary metabolites and secondary metabolites, a photosynthesis regulation, a morphologic responses, a transcription regulation, stress-induced gene regulation, and antioxidant defense. That in this we we learn about the ROS scavenging. ROS means reactive oxygen species. Now the phytohormone signaling, TAS, cytokines, gibberellins, ethylene, and abscisic acid. And now there are the a classification of secondary metabolites. The first is alkaloids. These in alkaloids, a compound present in hormones, carotenoids, pigments, sterols, latex, and essential oils. There are the main features of this. A group of major importance with more than 40,000 molecules. They are considered of importance for the survival of plants. They are insoluble in water and derived from the union of isoprene units. Its properties is anti-carcinosis and anti-ulysses and antimicrobial, etc. Now these are the other phenolic compounds, glycosides. Now there are the some there are the some of plant species and their functions. Now the production of secondary metabolites, the process of in vitro artificial media, a culture of cells for the large production of secondary metabolites. As the first is the selection of cell lines for high yield of SM, large scale cultivation of plant cell, and the third is media composition and effect of nutrients, and the fourth one is elicitor induced production of secondary metabolites, and fifth is environmental factor, and sixth is bio transformation using plant cell culture, and the last one is secondary metabolites, release of analysis and identification. In this diagram, I explained everything. Now the summary and indication. Then the techniques commonly applied for cell line selection are cell cloning, visual or chemical analysis, selection for resistance. They are not directly involved in the normal growth and reproduction of plants, but they are essential for now there are the abiotic stress and abiotic stress in plants. The first is abiotic stress. The abiotic stress we can study about the drought stress, salt stress, high temperature, low water or high water. Result and conclusion. Secondary metabolites often occurs in plants are subjected to stresses including various elicitor or signal molecules. Secondary metabolites plays a major role in the adaptation of plants to the environment and in overcoming stress conditions. Environmental factors like temperature, humidity, light intensity, apply of water, minerals, and CO2 influence the growth of a plant and secondary metabolite production. Drought, high salinity, and freezing temperatures are environmental conditions, and they are the abiotic stresses that cause adverse effects on the growth of plants and the productivity of crops. Plant cell culture technologies have been effective tools for both studying and producing plant secondary metabolites under in vitro conditions and for plant improvement.
Hello.
Ma'am, wait a minute. This slide is not working.
Okay.
These are the technical. These are the technologies we used in production of secondary metabolites and mechanism in plants. These are the microbiome microbiome. These are the data analysis and statically analysis and metabolite identifications. As I already told in the as I already told in the introduction, now the acknowledgement and references. I would like to say thanks my guide Dr. Kunal on this work, and especially thanks to the coordinator and teachers to give us such proudly opportunity for show my talent. These are the references. Thank you.
Thank you, Pritiman.
Thank you, ma'am.
Now the other presentation is by Deepika.
Yes, ma'am. Can I present now?
Yes, yes.
Okay.
Deepika, now you can start sharing your screen and can have
Yes,
your
ma'am.
presentation.
Just a minute. Ma'am, is it visible?
Deepika, are you available? A final call for Deepika for her presentation. Okay, seems to be Deepika is having technical issues. So this is all about the International Conference on Clinical and Applied Microbiology. I would like to thank each and every participant who have joined this conference for 2 days, that is on 26th and 27th and 28th April 2023. This is to inform all the participants that the winner announcement and the certificates for the presentation and participation will be given on their registered mail IDs by next week. I repeat again, the winners list and the certificates for the presentation and participation will be giving on their registered mail IDs by the next week. So with that note, we are closing the today's conference of International Conference on Clinical and Applied Microbiology. We are very glad to have you all here And we are very much delighted to see you all people in our upcoming conferences also, which we have been displayed the details previously. And if you have any queries regarding that, you can contact us by the next week. Bye-bye. See you all in our next conferences.
Ma'am, excuse me, ma'am.
दीपी
हाँ,
हो गई वो क्लोज हो गई वो
गए?
मीटिंग, अब नहीं इसमें कोई भी। हाँ
फिर अब
इसमें नहीं है, तू एक सेकंड रुक जा, मैं एक बार सुने, जो नंबर नहीं है, जो नंबर आ रहा है
हाँ,
उस
कौन सा
नंबर
नंबर आ रहा है?
अरे वो नंबर नहीं आया था एक व्हाट्सएप पे, मतलब जो मैम फोन करके बता रही है कि तुम्हारी है आपकी बार टर्न, रेडी रहना।
मेरे पास नहीं आया कोई भी नंबर शुरू से ही।
मेरे पास व्हाट्सएप पे नंबर आ रहा था किसी को बताने, हाँ तो मैंने उसको लिख के बोला कि मैम मतलब कि मैं मेन हॉल में दे दूंगी, तो उसने बोला कि हाँ
Hey
ठीक
girls, can you please mute the other participants as well? So it would be nice if you can have the conversation privately, if it's okay.